Good morning, dear colleagues, esteemed guests and friends. On behalf of Organizing Committee, I welcome you all to this special symposium on preclinical innovation and regulatory practices. This event is being organized to commemorate the superannuation of Dr. S.K. Rath, who has been truly a remarkable colleague and one of the most respected member of CDRI family. Today we have gathered here not just to bid him farewell, but also to celebrate his illustrious journey of 27 long years, which he started in year 1999. On this occasion, we are delighted to have with us Dr. G.N. Singh, Advisor to Honorable Chief Minister of Uttar Pradesh and former DCGI, as our chief guest. I request our Director, Dr. Radha Rangarajan, to please welcome him with the bouquet. We are equally honored to have with us Professor Alok Dhawan, Director, CBMR, who has kindly agreed to be the guest of honor for this event. I request our Director, ma'am, to please welcome him with the bouquet. We are also expecting Dr. A.B. Pant to be with us. He's not here at the moment. He'll join shortly. With this, I request our Director, ma'am, to give her welcome address to start the event. Thank you.
Respected Chief Guest, Dr. G.N. Singh, Advisor to Honorable Chief Minister of Uttar Pradesh and former Drugs Controller General of India, esteemed Guest of Honor, Dr. Alok Dhawan, Director, CBMR, Lucknow. We're expecting Dr. A.B. Pant, Chief Scientist, IITR, former HODs of the Division of Toxicology and Experimental Medicine, of course, Dr. Rath, current HOD of Toxicology and Experimental Medicine, whom we are also honoring today. Dear colleagues, students, friends of Dr. Rath, friends of CDRI, welcome. It's a pleasure to bring you all together for this symposium on preclinical innovation and regulatory practice. We're really honored by the presence of Dr. G.N. Singh. He, as you know, has been dedicated to improving and strengthening the Indian regulatory system. And he's also been a major champion for the development of the pharmaceutical sector in Uttar Pradesh. Thank you for accepting our invitation. We're equally delighted to welcome our guest of honor, Professor Alok Dhawan, Director of CBMR, and of course a long-standing friend of CDRI. He's a distinguished scientist, and his leadership and vision for translational research at CBMR are exemplary. I extend a warm welcome to all our colleagues, students, and participants of the symposium. Your presence here makes it very special, so thank you for joining us. Today's symposium is on preclinical innovation and regulatory practice. Why this particular combination of themes? Well, drug discovery is at the core of what we do at CDRI, and as we progress from hit to lead to optimized lead, our focus remains on potency, efficacy, and pharmacokinetics. But when we enter the phase of preclinical development, the gamut of toxicity, formulation development, scale-up, and stability studies are required. Compounds can fail at this stage, even though they may have shown excellent efficacy in the previous phase. Innovation to enable earlier de-risking so that cost and time can be reduced are key aspects of improving the drug discovery process. This entails good science, adherence to good practice, and promulgation of sensible policy. Today's talks will give us insights on all these aspects, given the diverse experiences of our speakers. But before we start the symposium, we have the first part, which is really dedicated to celebrating one of our very special scientists, Dr. S.K. Rath. He's chief scientist in the Department of Toxicology and Experimental Medicine, and as Smriti said, he'll be retiring after 27 years of dedicated service. So a little intro, or not intro, but I'll share some background information for those of you who don't know him as closely. Dr. Rath completed his master's from Uttar University in 1986 and then got a PhD in zoology from Banaras Hindu University in 1995. He was a postdoctoral fellow at CSIR-CCMB Hyderabad, and he joined CSIR-CDRI in 1998, becoming chief scientist in 2017. Over nearly 3 decades, Dr. Rath has made profound contributions to regulatory toxicology, molecular pharmacology, and environmental health. His scholarly impact includes more than 172 publications in prestigious journals such as Science, Cancer Research, PNAS, Human Genetics and Neurotoxicity, with more than 5,300 citations, a reflection of the enduring value of his research. His excellence has been recognized through multiple national and international honors, including the Genomic Pioneer Award in 2008, 2 CSIR Technology Awards, and a Raman Research Fellowship, which he availed of at the Johns Hopkins University. Beyond his individual achievements, one of his most enduring legacies is the establishment and growth of CDRI's NGCMA certified GLP facility, which is now entering the 8th successful year. This is a jewel in our crown, a very critical part of our ability to do drug discovery work. And as you know, we are one of a very small number of academic institutes in India that is able to sustain a GLP toxicity facility. This has required continuous dedication, adoption of process, cajoling and coaxing of people to follow documentation, to follow protocols, relentless monitoring, and much more. It is a lot of effort. So our deepest gratitude to you, Dr. Rath, for making this facility functional and productive and really an example of what can be done within an academic institution to further the cause of drug discovery. Thank you very much. I think what is clear about Dr. Rath's leadership style is that, he really leads by example. And so if he expected the team to work day and night to get the GLP facility ready, he was also very much in the front lines, rolling up his sleeves and doing the work. And that's a very, very special leadership style. Thank you. I also want to thank you for your remarkable contributions to institutional work. So I actually pulled out a list of committees that you've been part of. They're too numerous, but I'll name the major ones. The Translational Research Group, which you've been part of for years. The Monitoring Committee for the Phase 3 of study of Picrolive. The Laboratory Strategic Group or the CDRI Strategic Group. The In-House Project Review Committee. The Animal House Committee, Safety Committee. Year after year, you've been part of the CSIR Foundation Day Celebration Committees, the Annual Day Celebration Committees. You've been probably, a member of every major event committee that has been that has existed at CDRI. I couldn't find the entire list, but then you've also been part of the Purchase Committee, Investment Committee, Committee for Procurement of Specialized R&D Services, Standing Disposal Committee, Staff Welfare Committee. All this reflects that Dr. Rath was always thinking beyond himself and dedicated to the institution's causes. Institution building really is a very difficult process, and it takes people who can think beyond themselves and who can really offer their time and effort towards things that benefit the institution but not necessarily bring individual glory. In fact, the most recent example of his institutional work was the recruitment of junior stenos and JSAs. We had 14,000 applications. He chaired the committee and sailed this through just last month. And he never once came to me to say that, he's nearing the end of his tenure and would not like to do the work. That was the other remarkable aspect of Dr. Rath. If you gave him something or you asked him something, he was always willing and able to complete the job. So he became a highly reliable and highly dependable colleague. Beyond CDRI, Dr. Rath is a well-recognized expert for many of India's scientific bodies and for CDSCO. In fact, the DCGI asked about what would happen when you retired, because you're part of several SECs. And he has a remarkable network of friends and colleagues across institutions, both within and outside CSIR. It's a testament to his ability to network, to really care for people, and to be there for colleagues and friends and beyond colleagues, families, etc., etc. I think there'll be some interesting stories from colleagues later on. Dr. Rath has been an exceptional mentor. He has nurtured students, scientists, toxicologists, and really helped them succeed. Personally, I want to thank you for being a pillar of support and for offering always candid opinions on matters of the Institute. I've relied on you, and you've always helped, encouraged, and offered a balanced opinion. Thank you very much. On behalf of the CDRI family, I extend our deepest gratitude for your service and leadership, and I wish you a very active, healthy, and joyous life ahead. Once again, I welcome you all to this symposium today.
Thank you, ma'am, for such a nice welcome address. It's an honor to welcome our chief guest today, Dr. G.N. Singh. He's a towering figure in India's healthcare and regulatory landscape. Dr. Singh currently serves as advisor to the Honorable Chief Minister of Uttar Pradesh, where he continues to contribute in strategic guidance to health and pharmaceutical policy for the state. He is best known nationally for his impactful tenure as Drug Controller General of India, a position that he held with distinction for 6 years. During this time, he brought about transformational changes in the country's drug regulatory framework, ensuring greater uniformity, transparency, efficiency through simplification of rules and integration of technology. An alumnus of IIT BHU Varanasi, where he earned his B Pharma M.Pharma and PhD. Dr. Singh also holds MBA from University of Hull in UK. He was the founder director of Indian Pharmacopoeia Commission, India's apex body for setting standards for medicines, a position that he continues to associate it with even now. Over the years, Dr. Singh has been trusted as an expert and advisor to multiple national and international bodies, always known for his humility, integrity, and dedication to improve public health. Please join me in extending a very warm welcome to Dr. G.N. Singh while I invite him to deliver his address as chief guest of the event. Sir.
Good morning, ladies and gentlemen. It is a great honor and pleasure to stand in front of you. Whenever I get an opportunity to visit CDRI, I feel proud in myself. Today is a great day when we are going to facilitate one of our best scientists, Professor Dr. S.K. Rath Sahab, who has contributed so significantly. I am equally, when I was entering, few years back when I entered into the campus, it was not that lush green which I felt today, and I congratulate the director and its staff for maintaining so well. And we are very grateful to you for maintaining our campus so beautifully. I always remember my mentor, Dr. Nityanand ji, who is no more, but he always inspires us how the science and humility can go together. You people are very fortunate to see him, to work with him, and get his guidance from time to time. We are very fortunate to have another great person, my colleague, Dr. Dhawan Sahab, who is carrying the legacy from generations. I met his father, got his blessings, and equally I am fortunate to have his guidance on various issues for the development of Uttar Pradesh healthcare and in nation in totality. I remember in 2004 when GLP was the issue, the country was looking forwards, how GLP would be shaping in India. And I remember Dr. Tiwari ji, Dr. G.L. Tiwari ji, he was the man who rooted, D.S. Tiwari ji. D.S. Tiwari ji who rooted this GLP concept in this country, and we were the few people who attended the GLP inspectors meeting at Jaipur. So we were at that time GLP facilities were very rare, and the country was entering into the preclinical studies phase. We were thinking how to reduce the cost of the pre-investigational studies, plus how we are going to strengthen the government institutional system to be partner of that. And I'm highly delighted to know that GLP certified facility the CDRI has developed, and we congratulate its director, its team for doing such wonderful work. I remember Dr. Srivastav, who was at that time from this institute, and he is the man who made and educated the things to happen. Afterwards, the NIPER, many people came. Why I'm mentioning this thing, this institute has got a lot of legacy. In the present context, when I'm seeing young people sitting over here, you are very fortunate to have most of the things put in place. Now the globe is looking towards India, how from the generic producing nation, we are going to enter into the innovative phase. All the components have been put in place. Only it requires your standing and making the things to happen in this country. When we are going to see that by 2047, our Honorable Prime Minister has a vision, जब अमृत काल में प्रवेश करेंगे तो सबकी परिकल्पना है और हम लोग की इच्छा है कि भारत जो है एक विकसित भारत के रूप में आएगा. And healthcare sector is going to play a very significant role. दो लोगों का सबसे महत्वपूर्ण योगदान होगा, एक एजुकेशन सेक्टर का, दूसरा हेल्थ सेक्टर का. health sector में innovation is the key which is going to drive the healthcare sector of this nation. Not only of the nation, but at the global level. The whole humanity is looking toward the Indian scientists, how they are working, how they are inventing, how they are going to provide the services, and through their innovations to see that the one health of the globe could be properly addressed. तो हमारा आपसे यही निवेदन है कि जो CDRI इतना बड़ा विजनरी हमारे फोर फादर लोगों ने बनाया है, उसको आप लोग कर दीजिए कि अब आने वाले समय में जब पूरा विश्व हमारे तरफ देख रहा है, माननीय मुख्यमंत्री जी कल ही देखिए पहली बार उन्होंने हमारे चारों CSIR की इंस्टीट्यूशंस की प्रशंसा की थी। कल आपके माननीय मंत्री जी थे, माननीय मुख्यमंत्री जी ने जो ग्रीन डाटा हमारा बनने जा रहा है गाजियाबाद साहिबाबाद में, जो आपकी CSIR की जो प्रॉफिट अर्निंग हमारी कंपनी है, देखिए कभी उन्होंने यही बता रहे थे कभी घाटा कर रही थी, लेकिन आज जो अपने सबसे महत्वपूर्ण काल में प्रवेश की है। CDRI भी कई अप और डाउन्स देखी है, हमने देखा है कि कभी हम इन लोगों को बताएं कि कैसे हम CDRI से अटैच्ड है। कि हम जब छोटे थे तो हमारे फादर यहाँ पे आए हुए थे विधानसभा हमें दिखाने के लिए, कोई हम 7th 8th में पढ़ रहे थे, रिक्शे से हम यात्रा कर रहे थे और CDRI की तरफ से गुजर रहे थे, तो उन्होंने कहा कि ये भारत की सबसे महान संस्थान है, यहाँ की जो लैब्स हैं वो एयर कंडीशनड हैं, यहाँ के साइंटिस्ट पूरे विश्व में प्रसिद्ध हैं। तो वो हमें इंस्पायर हुए, हमने कहा कि नहीं, इट इज़ बेटर टू गो फॉर फार्मेसी। आई गॉट एडमिशन इन इंजीनियरिंग आल्सो, बट by seeing those facilities and inspired by my father showing that CDRI is situated in Uttar Pradesh, हमने कहा कि नहीं हम फार्मेसी पढ़ेंगे। और देखिए फार्मेसी पढ़ने का ये CDRI का एक बड़ा योगदान सबके लिए है। और जितनी न्यू ड्रग्स डिस्कवरी अभी तक भारत में हुई है, क्रेडिट गोज टू CDRI। मोस्ट ऑफ द मॉलिक्यूल हैव बीन इन्वेंटेड फ्रॉम हियर। वी आर लुकिंग फॉरवर्ड्स। देखिए डीप पॉकेट चाहिए, सारी चीज़ें चाहिए। हमारा आपसे निवेदन है कि लेट देयर बी प्रॉपर कोलैबोरेशन among the industry, academic institutions and the end users. How we are going to see that the blockbuster drugs could be produced from India. The whole globe is looking towards India. इन्हीं शब्दों के साथ अभी हम बाद में जब अपना प्रेजेंटेशन देंगे तो आपको बताएंगे कि उत्तर प्रदेश क्या कर रहा है, किस दिशा में जा रहा है, हाउ वी आर गोइंग टू प्रोग्रेस एंड वी आर गोइंग टू डेवलप यूपी एज ए फेवरेबल डेस्टिनेशन फॉर pharma sector, medical devices and health sector. With these words, I express my gratitude to director, all the people who are gathered over here, especially Dr. S.K. Rath साहब। इनको अभी रिटायरमेंट के बाद डॉक्टर साहब बड़ी जीवन अच्छा हो जाता है। आप ये बस सोचिएगा, आप ब्यूरोक्रेट नहीं हैं। देखिए ब्यूरोक्रेट रिटायरमेंट के बाद वो किनारे हो जाते हैं, लेकिन एक हमने जो अनुभव किया है, साइंटिस्ट जो है वो ग्लो करता है, उसकी इज्जत जो है बढ़ती जाती है। केवल एक ही निवेदन है कि आप एक्टिव रहिएगा हमारी मतलब CSIR की लैब्स के साथ, यूपी गवर्नमेंट के साथ और रेगुलेटरी सिस्टम के साथ, कि आने वाले समय में आपका जो नॉलेज है, जो आपका मुस्कुराता हुआ चेहरा है, ये भारत को महान बनाने में योगदान देगा। इन्हीं शब्दों के साथ हम आभार व्यक्त करते हैं।
Friends and colleagues, Dr. A.B. Pant has joined us. Dr. A.B. Pant is already here. He's Chief Scientist IITR and he'll be delivering an insightful talk later during the day. But right now I would request our director ma'am to please welcome him with a bouquet. So we now move to the next section. Friends and colleagues, it's an honor to introduce Professor Alok Dhawan, an eminent scientist and visionary institution builder, who currently serves as Director of Center of Biomedical Research in Lucknow. Professor Dhawan has previously held several key leadership roles, including Director IITR, Director CSIR-CDRI, and Founding Director of Institute of Life Sciences, Ahmedabad. With a PhD in biochemistry from University of Lucknow and honorary DSc from University of Bradford, UK, Professor Dhawan has pioneered several domains in toxicology, most notably nanomaterial technology. Please join me in warmly welcoming Professor Alok Dhawan to deliver his address.
अमित जी की भाषा में अपने नाम पे कसीदे सुने नहीं जाते हैं. बहुत बहुत धन्यवाद. सबसे पहले रथ साहब का, जिन्होंने हम लोग को ये मौका दिया कि सब साथ मिलके आपकी जर्नी को सेलिब्रेट करें, आपके मुस्कुराते चेहरे को और देख पाएँ हम. मैं समझता हूँ एक जो पूरा हॉल भरा हुआ ये दर्शाता है कि रथ साहब की जो पॉपुलैरिटी है वो न केवल अपने डिवीज़न में है बल्कि पूरे CDRI में है. आई थिंक what Radha mentioned in her speech, I think was very profound, that he has been able to contribute to the entire institute rather than just his division. And I think it's also true that he has been part of other institutions including IITR, where he played a stellar role in terms of GLP, in terms of lot of workshops that he came as a faculty member and trained people. I think I don't know how to put it, but I think in 2002 we started the CMM, CSIR mission mode programs. So he was the nodal scientist for alternate to animal models in toxicology, and Dr. C.M. Gupta was the director of and was heading that mission mode. And the terror that CMG had, everybody knew. So the only way you sort of had was to go through SK Rath and ensure that you don't get scolded while you're presenting. So I think, but he ran that program wonderfully. There were, I think, 5 institutions to begin with, right? And he had an arduous task of putting together the entire report every 6 months, send it back to CSIR, and to ensure that everything was in place. Then came the second NWP network projects of CSIR, where again he was the leader, and that was to validate those animal models that had been developed in the past 5 years, from 2007 to 2012. And again he took the leadership role and he ensured that everything was in place. I think whenever we had to make the presentation, we would always sort of meet him before that, so that he has the entire worldview of what is happening in the project, and he would present it before we do, and then it was life made easy for us. The other thing I've never seen him grumpy, and never seen him scolding anyone. I think always had the smile on his face. So I think that nature of his ensured that whatever task was given to him was completed well in time and with no hard feelings. जब इनको बहुत गुस्सा आता था तो बस वो फ़ोन उठा के बात कर लेते थे, ख़त्म हो जाती थी बात. आई थिंक बट एन एंड ऑफ द डे आई थिंक रथ इज़ व्हाट इट इज़. रथ हैं ये, रथ यात्रा भी आज है. सो आई थिंक रथ हैं तो रथ सबको अपने साथ ले जाता है. सब लोग रथ को घसीटते हैं, रथ सबको घसीटता है, वॉट एवर इट इज़. बट आई थिंक बट एंड ऑफ द डे ही पुल्स इट ऑफ. यू नो, आई थिंक दैट्स द रिमार्केबल थिंग अबाउट SK. And as you rightly mentioned, he is not only part of CDRI, he has a huge network of people in different institutions. He's kept in touch with his alma mater. So whenever you talk of BHU, they talk of him. You know, he's done really well on that account as well. On his family front, he's done wonderfully well. And one thing I don't know if you guys noticed, the first responder, if I may put it that way, of any program happening here is SK Rath. So as soon as the program starts, the first photographs will be posted on Facebook. ये आ गए हैं, ये शुरू हो गया है. And today, for some reason, not clicking photographs, but that was how we came to know that something is happening in CDRI. So that only shows his love and affection for the institution, where he spent, you know, almost half of his life, active life, I would say. And it also shows, you know, that how much he adores what happens, irrespective. जिसका भी सेशन हो रहा हो, जिसका भी लेक्चर हो रहा हो, आपका पोस्ट पहले आ जाता था फटाक से. इन्होंने ये बोला, ये पोस्ट हो रहा है. आई थिंक जीएन सिंह साहब ने बताया लोगों के बारे में जिन्होंने CDRI को यहाँ तक पहुँचाया है. इसमें कोई दो राय नहीं है कि CDRI को बहुत लोगों ने नर्चर किया है, बहुत लोगों ने उसको अभिसिंचित किया है यहाँ तक पहुँचाने के लिए। और ना केवल ये, मैं समझता हूँ जैसा आपने कहा, हम आगे भी इस विषय पर चर्चा करेंगे, लेकिन भारत सरकार ने भी CDRI को बहुत रिस्पॉन्सिबिलिटीज़ दी जो कि उन्होंने बखूबी निभाई। और जब हम प्री क्लिनिकल इनोवेशन की बात कर रहे हैं, रेगुलेटरी प्रैक्टिस की बात करें, शेड्यूल वाइज CDRI और IITR ने मिलकर लिखा था। So I think that is the profoundness or importance of these institutions that have actually contributed to the policy of this country where we are today. And Dr. G.N. Singh rightly said he is also beneficiary of this institution. जब Pharmacopeal Commission बना तो Dr. Nityanand Saab has played a stellar role in that as well. And the first beneficiary of that was Dr. G.N. Singh. So I think during the day we'll not only celebrate Dr. Rath, but of course what he has been able to achieve in the precincts of CDRI, which has not only nourished and nurtured scientists like him, but has actually nourished and nurtured so many CEOs or heads of vice presidents or heads of departments of various pharmaceutical companies in the country and abroad. There are at least 2 CEOs that I can remember offhand, Dr. Sanjay Singh and Keshav Dev, who have their own companies. I can't remember, to be honest, other names who are currently CEOs, but at least senior vice president and vice president from So I think it's commendable that how institutions that were built with a vision have actually served the purpose for this country, and how people like Rath have actually ensured that the institution moves on with new things in place. GLP being one of them. I think Dr. Pant is here. He'll also perhaps speak about that as well. I don't know. But yeah, I think the friendship that I've enjoyed with Shrikant has been exemplary. A lot of anecdotes. I don't know if I should share them here. He still has a couple of days to go before he's superannuated, so I don't know if any paper would get hampered by me speaking here. So, yeah, but I think Shrikant, congratulations for this wonderful journey in CSIR, and on behalf of CSIR and my own self, I would like to congratulate you and your family for dedicatedly giving half of your active life to CSIR, of course. And we look forward to exciting times for you ahead. So thank you very much indeed. Thank you. Namaskar.
Thank you, sir. With great pleasure and a sense of nostalgia, now we move to the next section of the event, where we celebrate life and career of our dear colleague, Dr. S.K. Rath. Many of us already know that he is a stalwart in the field of regulatory toxicology, but beyond his professional accomplishments, Dr. Rath has been an exceptional mentor to many of us, always willing to share his knowledge, offer advice, and encourage us all. He has been a troubleshooter for many of us next generation scientists. And let's not forget his exceptional sense of humor. Whenever he's in a room, he lights up the room, and then whenever there is a stressful situation, his comments and his jokes often lighten up the environment. He's a remarkable human being who has touched all our lives in a meaningful way. And with that, I now invite Dr. Rath's friends and colleagues to share their reflections and reminiscences about the time that he spent with us and with them. I would like to begin with Dr. Sudhir Srivastava, who was heading our division back then when I joined. So please.
Good morning to all of you, and I'm lucky to be here and sharing these moments with Dr. Rath's lifelong journey and with this turning point in his life, which is called now as the watershed moment for him. बहुत ही अच्छा था. Whenever I could think of Dr. Rath, he was always a well-behaved person. Still he continues to be, and whenever I make any request to him, even at this stage, I have been out of CDRI for the past 17-18 years now. But he joined here during the time in 90s when the Department of Toxicology was going through a phase of metamorphosis. And he was the lucky one to pick up to add all the full range of toxicity studies. Before he joined, we were doing only AIMS test and probably micronucleus test, which was standardized by Dr. A.K. Giri. But Rath gave the full complement, full panoply of all the in vitro, in vivo tests of regulatory toxicology studies. Not only that, he simultaneously encouraged the research part in the division of toxicology. So with all that in mind, I think he has been a remarkable human being also, and she has already spoken about the sense of humor that he has. I will just say a small thing which he once remarked, and we were all having, as heads of the divisions, we used to have our weaker moments also, when something was going against our wishes or something happening inside the department, not coming to our own expectations. And he gently said, "Sudhir sir, every department has been allotted with its own problems in a different way." And I still remember that. It really cheered me up. Every department has some or the other nuisance brewing here or there, and we have to we all have to come to terms with it. So with that, I wish him all well, and his family, his children. बहुत अच्छा आपको लगा, मुझे भी लगा कि आपको मैं इस समय join कर सका. I've been into a different phase of my life now, but still I feel like homecoming whenever I'm here sitting with the scientists here. I remember all those very good things that happened, that taught me many things also in the process. So once again, best wishes, and thank you, madam, for inviting me here.
May I now request Dr. Nath to share his experiences with us?
आप सभी को नमस्कार। सर्वप्रथम तो मैं श्रीकांत को इनके एक गौरवशाली सराहनीय सेवा काल संपन्न करने पे बधाई देता हूं, क्योंकि कोई भी वैज्ञानिक सेवा काल निर्विघ्न संपन्न हो जाए, यही बहुत बड़ी उपलब्धि होती है। मैं भी रहा हूँ 37 साल, मैंने भी सर्व किया है। तो इनके पीछे तो कहा गया, ये मेरे सहयोगी मित्र मात्र है और हमेशा बहुत अपने काम के लिए समर्पित थे और जुझारू थे। सबसे बड़ी चीज़ क्या है, इन्हें आपको एक काम दिए, चैलेंज दीजिए, जो आपके करने हिम्मत है, ये स्वीकार करते थे सहज और उसके लिए लग जाते थे और सब चीज़ें सफलता करके दी। उसका सबसे उदाहरण, जैसे सुधीर ने कहा, जीरो टॉक्सिसिटी लैब के एस्टेब्लिशमेंट, ये मतलब भारत में एक मापदंड बन गया था यहाँ के CDRI का ही है इसका। और दूसरा, जैसे एक इन्होंने कोट किया, इनका प्रॉब्लम वाला, इनका एक कोट मुझे भी याद है, जब मैं डिपार्टमेंट ज्वाइन किया शुरू में, तो डिपार्टमेंट तुम भी आते, इन्होंने कहा था अलॉटमेंट ऑफ प्रॉब्लम्स, तो उस पर इन्होंने बहुत बढ़िया कहा था कि साहब मतभेद हो सकते हैं, मनभेद नहीं होना चाहिए। श्रीकांत का बहुत फेमस कोट था, वो मैं लोगों को सुनाता था, देखो ये डिफरेंस ओपिनियन होती हैं बट मन में कोई ओपिनियन, ये श्रीकांत ने कहा था। और मैं अब इनको इसमें कि ये हमेशा इन्होंने एक नई चीज़ किया है, प्रेरणा स्रोत रहे हैं, तो अपने रिटायरमेंट के बाद भी ये लोगों के लिए एक इसका लोगों में उत्साह बढ़ाएंगे और एक इंस्पिरेशन नई जनरेशन देते रहेंगे, और CDRI को जो भी भाव होगा उसको ये पूरा करते रहेंगे इसमें। और इसके साथ ही मैं श्रीकांत और इनके परिवार को एक सुखद जीवन की, सर्वाधिक सुखद जीवन की शुभकामनाएं देता हूं और आशा करता हूं कि इनका इंटरेक्शन बना रहेगा, क्योंकि रिटायरमेंट बाद भी हमेशा हमसे इंटरेक्शन में रहते थे श्रीकांत। और दूसरी एक चीज़, और CDSO में CDRI की प्रतिनिधित्व की एक परंपरा रही है और जिसको इन्होंने बखूबी निभा रहे हो ना, अभी SCG में चल रहे हैं, तो ये एक चीज़ इन्होंने ये बड़े सराहनीय ढंग से की। और आज भी CDSO में CDRI के नाम देने वाले हैं तो श्रीकांत रथ हैं। और मैं एक अवसर एक और लेता हूँ इस मतलब, मालूम पड़ा मैं पुराने और मित्र सहयोगी अमित भी इनके साथ ही जा रहे हैं, तो भैया अमित, तुम्हारे लिए भी मेरी बहुत शुभकामनाएं, तुम्हारे परिवार के लिए, और तुम भी CDRI को अपनी देते रहा सेवा, क्योंकि इन्होंने भी बहुत, ये भी बहुत ही समर्पित, क्योंकि जब मैं यहाँ पे एरिया कोऑर्डिनेटर ड्रग डेवलपमेंट था तो अमित का उसमें बहुत बड़ा रोल होता था। हाँ, तो इसको भी मैं इस अवसर का इनको शुभकामनाएं, बधाई देता हूँ। और रथ और संपर्क बना रहेगा रथ से, मतलब ये नहीं कि आप सक्रियता से अलग हो गए। ओके, धन्यवाद।
We are also joined by one of our former head of department. Dr. R.K. Singh. I request sir to please come on stage and share his memories with us.
फ्रेंड्स, डॉक्टर रथ साहब ने टॉक्सिकोलॉजी डिपार्टमेंट 1998 में ज्वाइन किया था। उससे पहले 28 फरवरी 1985 तक ये डिपार्टमेंट ऑफ टॉक्सिकोलॉजी एंड एक्सपेरिमेंटल मेडिसिन था। डॉक्टर नित्यानंद साहब जो हैं वो 31 दिसंबर 1984 को रिटायर हुए थे, और डॉक्टर एस.के. मुखर्जी जो कि 20th सेंचुरी में ही वॉज हेड ऑफ टॉक्सिकोलॉजी एंड एक्सपेरिमेंटल मेडिसिन डिपार्टमेंट, ही सुपरिन्यूएटेड ऑन 28 फरवरी 1985। 1 मार्च 1985 को 2 डिपार्टमेंट बना दिया गया, एक टॉक्सिकोलॉजी बनाया गया और एक क्लिनिकल एंड एक्सपेरिमेंटल मेडिसिन बनाया गया। और 1 जुलाई 2017 को फिर ये 3 डिपार्टमेंट मर्ज कर दिए गए, टॉक्सिकोलॉजी, क्लिनिकल एंड एक्सपेरिमेंटल मेडिसिन, बायोमेट्री एंड स्टैटिस्टिक्स। Then again it became toxicology and experimental medicine. हमने 1983 में CDRI ज्वाइन किया था। उस समय डॉक्टर नित्यानंद साहब डायरेक्टर थे, और जीनोटॉक्सिसिटी डॉक्टर R.N. कार साहब देख रहे थे, और जो सारे टेस्ट से दे ऑल वे आर डन इन टॉक्सिकोलॉजी एंड एक्सपेरिमेंटल मेडिसिन। नेक्स्ट ईयर ही टुक ट्रांसफर टू Bhubaneswar, and there was a gap of I think 3 years. Then Dr. A.K. by that time genotoxicity was almost stopped. When Giri came, then again he started, and also he took transfer and he went to Kolkata. When Dr. Rath came in 1998, he established all the tests required by different guidelines, different regulatory authorities, और हम लोग सब मिलकर के 1986 में डॉक्टर श्रीवास्तव साहब आए थे। सब लोग मिलकर के, वो मेरे कोलीग भी हैं, और हम लोगों ने, सब लोगों ने मिलकर के टॉक्सिकोलॉजी को इस लेवल पे पहुंचाया कि जीएलपी डॉक्टर श्रीवास्तव साहब ने स्टार्ट किया था, बहुत मेहनत करी, और लेटर ऑन डॉक्टर रथ और डॉक्टर शरद शर्मा और आईआईटीआर के सारे कोलिग्स, क्योंकि 2008 में वो आईआईटीआर हो गया था, 1 जनवरी 2008, उससे पहले वो आईटीआरसी था। डॉक्टर रथ साहब ने मिल करके और सब ने मिल करके टॉक्सिसिटी का जो प्रीक्लिनिकल इवैल्यूएशन होता है, क्योंकि पहले हमें याद है कि उस समय 19 डॉक्टर एम धर साहब थे, तो उन्होंने ये कहा था, मुझसे सवाल पूछा था मेरे इंटरव्यू में, हाउ मच टाइम विल यू टेक इन टॉक्सिसिटी कंप्लीशन ऑफ ए कंपाउंड? आई सेड 3 years. He said that till now it is up to 10 years. And you must be knowing one of the compounds, Sancroman. History में Sancroman की toxicity was for the longest time. अगर आप देखेंगे Sancroman's toxicity was done many times, and it was cleared in I think 1988. It came in the market in 1988, and it was invented by Dr. Nityanand in 1967 or 68, and after a gap of 20 years. Now you see, when a compound comes to a department, one must ensure that it should be completed within less than 3 years. What is the routine nowadays in different countries? That they are taking a prestigious company, Himalaya Drug Company, where they are having the toxicity in Bengaluru. That is the only lab who is having preclinical toxicity evaluation of herbal products. Because herbal products, they also need the preclinical toxicity evaluation. And their promise is to put one product every year, one herbal product. And they are getting permission. And today Himalaya is supplying their products to more than 150 countries. And they are having for that they have made their own cultivation of plant center in Bangalore. More than I think 200-300 acres. So that type of research and innovation is needed today. और डॉक्टर रथ साहब का मेरा साथ बहुत अच्छा रहा है और हम लोग साथ में खूब एंजॉय करते थे, एक दूसरे से आजकल की चीज़ें डिस्कस भी करते थे. जैसे हमारे डॉक्टर श्रीवास्तव साहब हैं, डॉक्टर नाथ साहब हैं, हम सब लोगों का इतना बढ़िया ग्रुप था और अभी भी है. जितने नए लोग साइंटिस्ट ऐड हुए हैं सब लोग अच्छे से पढ़ते थे, सब लोग एक दूसरे से जो पढ़ते थे. इस तरह का माहौल होना चाहिए जैसे यूनिवर्सिटीज़ में है और इस तरह का माहौल बहुत अच्छी चीज़ है. और साथ में मैं डॉक्टर अमित मिश्रा साहब को उनको भी बधाई देता हूँ कि ये मैंने पूछा कि आपका रिटायरमेंट कब है तो उन्होंने कहा मंडे को. कहा जिस दिन डॉक्टर रथ और हम दोनों ने एक ही दिन ज्वाइन किया था. तो हमने अपने मन में सोचा एक दिन ज्वाइन करने का मतलब थोड़ी है कि एक दिन जाए जो है, भाई आगे पीछे भी हो सकते हैं. हो सकता है कि भाई कोई ऐसा प्रोजेक्ट बना के भेजते डायरेक्टर के थ्रू, ही मस्ट हैव दोनों को कुछ ना कुछ. लेकिन अच्छा ये दुनिया में एक दिन जो आया है तो जरूरी है एक ही दिन जाए जो है. लेकिन मैं उसमें नहीं जाना चाहता. मैं सिर्फ ये करना चाहता हूँ कि देखिए लोग ये सोचते हैं कि देखिए अभी श्रीवास्तव साहब ने कहा कि लोग ये सोचते हैं कि जिस दिन रिटायर हो जाओ CSR से पीछा छूटे बहुत खुश होते हैं. जीएन सिंह साहब ने भी इंडिकेट किया था. लेकिन एक बात आप याद रखिएगा, जिस दिन आप CDRI से रिटायर होकर चले जाएंगे उस दिन आपको जाते थे जो है एक्सपर्ट होकर के एग्जाम वगैरह लेने बायोकेमिस्ट्री का वो सब जो है वहाँ बताया. उस PGI को मैंने साल भर के अंदर यूनिवर्सिटी बना दिया है, SBS यूनिवर्सिटी. और वहाँ पे मैंने 11 स्कूल बनाए इंक्लूडिंग मेडिकल कॉलेज. और मैं 4 साल वहाँ पे वाइस चांसलर रहा. उसके बाद फिर मैं इंदौर, वो कह रहे थे अमित मिश्रा जी को कि आप तो बड़े लीन एंड टीन हो गए हैं. तो योगा इसीलिए करते हैं. आजकल देखिए प्रधानमंत्री जी ने अंतर्राष्ट्रीय योग दिवस मनाना शुरू किया है. ये नहीं कि यहाँ से रिटायर होकर के फिर खा करके फिर और मोटी तोन निकल आई, वो सब नहीं गया. इसलिए इन्हीं चंद शब्दों के मैं बोलना तो और भी चाहता था, लेकिन मैं डॉक्टर राज साहब अपने फोन की तरफ इशारा कर रहे हैं तो मैं उनको तो नहीं कहूँगा क्योंकि रिटायरमेंट के समय ऐसी बातें नहीं कही जाती. उसके बाद वो खुद ही करेंगे. इसलिए वो और उनका परिवार बहुत ही स्वस्थ रहे, प्रसन्न रहे और हमेशा CSR से जरूर जुड़े रहे क्योंकि CSR हम लोगों का ग्रैंडफादर रह जाए. इसी सब शब्दों के साथ मैं उनको और उनके परिवार को बहुत बहुत बधाई देता हूँ. धन्यवाद.
So after hearing things out from the head's perspective, I will now request Dr. Saman Habib to come and share her experiences. She has been contemporary to Dr. Rath and I hope she will give good memories to you.
Very good memories. Smriti is a little concerned about what I'll say. It's a real pleasure to be here, Shrikant, and I claim this special privilege because I am part of the group of scientists who joined at the same time. We joined within months of each other, and I think in the group of 5 or 6 of us, I'll be the only one still hanging around for the next few years. So that's not a good. It's not very pleasant for me to say goodbye. Yeah. So I want to say 2 things. Many things have been said about him and his immense contributions to the institution, to the department, to setting up systems. But you know, when all of us joined CDRI, it was, as was mentioned, in a phase of transition, and it was a strong institute in very traditional ways of working in drug discovery. But there were new things happening in the world which had still not been sort of established, taken in, and set up at the institution. And so, even though when we were hired, even though we were not told very specifically that this was our sort of brief, we were asked to set up our own experimental systems, our own labs, our own programs, and given a lot of freedom by Dr. C.M. Gupta. But I think at the back of our mind, all of us knew that we were were here for a purpose, that we were asked, we were supposed to do things for an institution and set up certain specific aspects of the way it was going forward and the way these systems had to be established. For Srikanth, of course, it was setting up specific toxicology systems apart from his research program. For Amit, it was drug delivery systems and formulations. For BN, it was something else. For me, it was setting up molecular and cell biology platforms and specific programs for research and so on. So all of us tried to do all this. But you know, apart from the science, I think it was also very important that we had a support system in the institution. We had very nice colleagues to work with. We had a very supportive administration initially, although I know, and all of us had very bad experiences as well, which is not the time to talk about it. Things have changed immensely since then. The institution has become a very collegial place to work in. We have a very nice environment. I know Srikanth has a big contribution in that aspect of the institution. But one thing I want to talk about, and because students are here, at that time there were PhD students in in fair numbers, but we didn't have a standard PhD program. So that students really suffered. They had to go to universities outside the CSIR system. They had to go to Kanpur or Awadh and so on and so forth. Register there, had massive problems in vivas, exceptional delays. It was a very bad system. And Dr. C.M. Gupta then tasked us, Srikanth, myself, Dr. Subramanya, so many others, to actually set up we were very young. I had just finished my PhD a year ago, and to set up the PhD program at the institute. And he tied up with JNU. Dr. Ashish Datta was the vice chancellor, who then said that he would register the CDRI with JNU, which was a big thing at that time. And we were tasked with setting up the PhD program. And I remember, setting up the coursework, which was not happening earlier, the pre-PhD program. It became a real solid foundation for the PhD program of CDRI to flourish. And in that, also Srikanth had a very major role to play. And the negotiations with JNU, presentations in JNU, setting up the coursework, getting it approved from JNU, it is a very stringent system. And it is because of that strong PhD program, that the transition for CDRI when ACSIR came up many years later was so smooth, because we had all the systems in place. And we didn't have any problem when we shifted from JNU to ACSIR. And for that, Srikanth also had a very important role to play. So that's, and for all of us in this sort of group of people who joined together, there is an understanding, I think, never said, never stated, that we all do our own thing. But if there is a problem of any kind, we are all there for each other. And Sanjay also is part of that group, right? So when all of you retire, and I know in the next 2 months, Sanjay is also going to say goodbye, I will not know who to call. And that is something that I'll really miss. So good, job well done, all the best. See you.
I will now request Dr. Amit Mishra to come and share his experiences. He does not like to hear about himself. I'm sure he'll talk a lot about Rath sir.
पहली बात तो ये है कि मैं इससे बड़ा हूँ। लोग बाग अपनी डेट ऑफ बर्थ गलत लिखवाते हैं, जिसमें उनको रिटायरमेंट के पहले थोड़ा टाइम मिल जाए। इसकी बर्थ 3 महीने बाद, पता नहीं क्यों ये जून में रिटायर हो रहा है। एनीवे, हमने कल भी कहा था, फिर कहेंगे, हम लोग को जब हायर किया गया था तो ओल्ड बिल्डिंग में श्रीकांत और मैं से BN तब तक आया नहीं था, समन थी वहाँ मीटिंग आई थी, और मैं फरहत बख्श पैलेस के पोर्टिको में खड़े होकर सिगरेट पीता था, और श्रीकांत BHU की ट्रेडिशन के हिसाब से बाहर जाके मुन्ना के ढाबा के पास से पान खाता था। तो हमारे बहुत से वरिष्ठ कोलीग थे जिनको ये बात बिल्कुल पसंद नहीं थी कि साइंटिस्ट को ये शोभा नहीं देता। तो उन्होंने कंप्लेन किया कि CM गुप्ता ने 2 लौंडे हायर किए हैं, एक पान खाता रहता है, एक सिगरेट पीता रहता है। तो वो तब तक लेगेसी थी, मतलब BHU। उसके बाद जैसे समन ने कहा, सीरियसली श्रीकांत ने अपने पर्सनल करियर के कॉस्ट पे इंस्टीट्यूशन बिल्ड किया। जिस टाइम को वो मतलब टॉप नॉच स्मॉल बायो वाला बंदा है या कैंसर ऑन्कोलॉजी में काम करने वाला, इसने सब छोड़ छाड़ दिया और इंस्टीट्यूशन बिल्डिंग के लिए जिसने कंट्रीब्यूट किया है, आई विल से संजय है, BN है, मैं हूँ, समन ने तो खैर किया है, BN ने किया है, संजय ने तो खैर बहुत बड़ी चीज़ की है। जो CVRS जो लगा हुआ है वो संजय की जो देन है यहाँ। वो इंस्टीट्यूट के लिए बैकबोन है, उसके बिना इंस्टीट्यूट चल नहीं सकता, वो संजय ने एस्टैब्लिश किया। तो हम लोग के ग्रुप के पास ये एटलीस्ट अपॉर्चुनिटी थी और सपोर्ट था लीडरशिप का कि हम लोग ये कर पाए। और सबसे ज़्यादा अगर मैं कहूँ तो श्रीकांत ने कंट्रीब्यूट किया, नो कॉन्स आउट सेइंग दैट। सो हैप्पी बर्थडे इन एडवांस 3 महीने एंड ऑल बेस्ट फॉर हैप्पी लाइफ अहेड। और पेंशनर्स एसोसिएशन में ज्वाइन कर रहे हो? मैं भी कर रहा हूँ, वहीं मिलते हैं।
May I now request Dr. Sanjay Batra to please share his experiences with us?
रहता था और मैं ऑलमोस्ट लाइक 2007 तक बेंच पे बहुत ज़्यादा काम करता था, सुबह से शाम तक। तो मैं बहुत ज़्यादा लोगों से बहुत मिलता नहीं था और ऐसी रिस्पॉन्सिबिलिटी भी नहीं थी। हालांकि 2003 में जब परचेज कमेटी में पहुंचे तो बहुत कुछ तब ज़्यादा लोगों का से मिलना जुलना होता गया। और उसके बाद व्हेन आई एस्टैब्लिश्ड माय ओन ग्रुप आई स्टार्टेड इंटरैक्टिंग। श्रीकांत, एवरीबडी हैज सेड दैट श्रीकांत वर्क फॉर इंस्टीट्यूशन बिल्डिंग, खाली इंस्टीट्यूशन नहीं। आपको पता है अभी इन्होंने IGIB का भी जो ग्रुप 2 ग्रुप 3 का रिक्रूटमेंट इन्होंने पूरा कराया है। एंड दैट्स बिकॉज पीपल बिलीव इन यू, दे गिव यू द रिस्पॉन्सिबिलिटी एंड यू हैव टू कम थ्रू दैट। ऐसा नहीं होता कि आपको बीच में छोड़ देना होता है या बीच में आपने छोड़ दिया। सब लोग आप पे इसलिए इस बिलीफ में आपको रिस्पॉन्सिबिलिटी दी जाती है कि आप पूरा करेंगे। एंड आई थिंक हमारे जितने भी यहाँ पे डायरेक्टर रहे उन्होंने श्रीकांत को जो भी रिस्पॉन्सिबिलिटी दी, श्रीकांत ने उसको पूरा किया। दैट्स अ बिग थिंग। एंड अभी अमित ने कहा श्रीकांत का बर्थडे 13 अगस्त को होता है और ये पहले फेसबुक पे हम लोग पता है कि 13 अगस्त है, लेकिन यहाँ गुस्सा नहीं देखा मैंने, देखा श्रीकांत का गुस्सा अभी हुआ क्या था कि हम लोग SDC के मेंबर हैं और श्रीकांत चेयरमैन है SDC का। और श्रीकांत की PIR में एक इंस्ट्रूमेंट था PCR जो कि इनको ट्रांसफ़र किया गया था और शायद वो उसकी उस समय कॉस्ट रही होगी 25 लाख या जो भी था, वो लिखा हुआ वहाँ पे। लेकिन वहाँ पे कुछ मेंबर्स जो है उसको बिलीव करने को तैयार नहीं। अकाउंट्स और एडमिनिस्ट्रेशन तो बिल्कुल खिलाफ में था कि ये कैसे ये है वो है। हालांकि फॉर्चुनेटली शरद वहाँ आ गए, उन्होंने जिसने बेचा था उससे भी फ़ोन करा के बता दिया। शरद उस दिन श्रीकांत बहुत गुस्सा भी हो गए थे, फिर हम लोग ने खैर पैसे। लेकिन देखिए गुस्सा भी इनका है, जैसे आपने थर्मामीटर लगाया चढ़ गया, उसके बाद थोड़ी देर बाद वापस आ गया। ये बाहर गए, शरद ने कुछ समझाया, वापस आ गए, क्या दैट वाज सॉर्टेड आउट। सो ऑफ कोर्स फ्यू थिंग्स समटाइम्स डोंट गो योर वे, you get agitated and you get angry about things, but coming back to normal within few seconds is something which Shrikant could do. I may not do that like that. And of course हम लोग ने कोर्स वर्क और ये सब जो बोला श्रीकांत, आई थिंक एकेडमिक यूनिट का पार्ट रहा है since we started coursework in I think 1999 2000 we started the coursework. And Shrikant has been part of academic unit all the time, either as main or as assisting things. And श्रीकांत की बहुत सी खूबियाँ हैं और श्रीकांत को मैं हर समय वैसे टोकता रहता था कि अरे पेट तुम्हारा बहुत निकल रहा है, समोसा कम खाया करो। वैसे ही वॉज ईजी गोइंग, मस्त रहता था। तो आई थिंक श्रीकांत की बहुत खूबियाँ। और जैसे सब ने कहा ये खाली BHU से नहीं मतलब जुड़े हुए हैं अभी तक, ये अपने स्कूल के और जो जहाँ ये उड़ीसा में जहाँ से इन्होंने स्कूलिंग की थी वहाँ से भी जुड़े हुए हैं। और ये संडेज़ को अक्सर ऑनलाइन मीटिंग करते हैं उनके साथ और फिर वो हम लोग को पता चल जाता है, क्योंकि जैसे आलोक ने कहा कि हम उसी वक्त फेसबुक पे आ जाता है तो वो पूरी गाथा फेसबुक पे आ जाती है, तो हम लोग को पता चल जाता था। सो वी कम टू नो मैनी थिंग्स एंड वी कम टू नो वेयर श्रीकांत इज टुडे। Either he is visiting Orissa or he is visiting some place, IGIB or CCMB or wherever he is, we come to know that okay, he's there, he's making things happen. So thank you, Shrikant, for your all the inputs, all the assistance, all the friendship you have been part of CDRI, and we wish you a very fruitful सेकेंड जर्नी. और ऑफ कोर्स ऐसे लोगों को कोई छोड़ता नहीं है, एडमिनिस्ट्रेशन भी नहीं छोड़ता है, लोग भी नहीं छोड़ते हैं. जैसे जीएन सिंह साहब ने कहा कि सीडीएसको के ये पार्ट रहे हैं. ऑफ कोर्स पहले हम लोग के यहाँ से कोई ना कोई सीडीएसको में हर समय रहा है. तो इस वक्त श्रीकांत थे और आई थिंक ही विल बी कंटिन्यूइंग एंड पीपल विल कैच हिम for various reasons, and that's for various good reasons, and that will be continuing. और श्रीकांत के एक बेटा और एक बेटी, and they have done exemplary, they are very good in studies and they have done very good, and we wish him a very healthy, fruitful and happy retired life onwards after 30th. Thank you.
As I told already, Dr. Rath was our go-to person for many of the new scientists which we joined much after him. He was our go-to person for whatever trouble we faced in finance or in purchase or wherever. And one of my colleagues, Dr. Ritu, we are almost contemporary, and we have had that kind of a help and support from him all along, even until now. And I request, we also had a tea club in Munna Dhaba Club in old campus. So these are friends from old times. I request Ritu to come here and share the memories of those times with the house.
Okay, so I think everything has been said about Rath, and I don't want to touch on his scientific accomplishments that he's had. I'll just talk about Rath, the person, the Rath. And I came to know the Rath, I think 21 years ago, Rath, and that was through BN. And BN is getting emotional about it. So our department and microbiology was just across, and I used to go to BN because I just joined new, and जब हम नया join करते हैं हमारा खून बहुत गर्म होता है, और जब चीज़ें नहीं हो रही होती हैं, we really don't know what to do about it, and I used to go to BN for instrument take इंडेंट करना है, ये कैसे करना है कि इसी कंपनी का मिल जाए, kind of things. And also about science. But then very soon after that, there was a group that was there, I think BN, Kamal and Rath, that used to go to Munna Ka Dhaba. And BN introduced me to this group कि चलो शाम को चाय पी के आते हैं. So it became 4 of us who used to go to, and slowly it expanded also, the group expanded, many of us joined Munna Ka Dhaba group over there, and that is how I came to know this rare species in CDRI, the Rath. He can say anything, anytime, in front of anyone. And so he would obviously, we all became friends, and you can see the standard deviation of friendship that Rath holds. I mean, I'm much down the line, but you can see all of us spoke about him, and he has even, the standard deviation even goes down. So, and everybody holds a little bit of Shrikant in them, and this is this thing that I had with Shrikant. And he slowly introduced me to the BHU group, both of them, BN and Rath, and the cell biology group. And not only did he introduce, but he knows everything about everyone to any extent. And I want to prove this. एक photo लगाइएगा. I wanted to send this picture to Shrikant in the morning. This is Shrikant back in 15, 16? Munnar, yeah. So as I said that he has, his hard disk keeps increasing all the time. This is how he tells stories about everyone. And he has kept names for everyone in CDRI besides their own names. I'm not going to reveal those names. Everybody has some other name than what they are in CDRI. So he introduced me to this BHU group, and he would, not only did he introduce me, he would tell me, I mean all of us, that how Professor Mercy was merciless, how Madhu's husband won the elections and the role of Shrikant in that. Not only did he tell me, but I met Madhu's husband at a BHU meeting, in cell biology meeting that we had. And then there came a promise that we will attend these meetings ever after. So we would all attend these meetings, these cell biology meetings very religiously, and BN would take the charge of putting the bills together, going, coming, everything. We would all forward it to BN. And there came a point where somehow all of us would be in the same committees. And it came into knowledge that, अरे इस committee के तो सारे लोग गायब हैं. They are all cell biology में गए हुए हैं. So we didn't know what to do. So there was a pre-meeting held before the cell biology meeting, where it was strategized कि 2 लोग पहले अप्रूवल लेंगे, 2 लोग बाद में अप्रूवल लेंगे, 1 जन पहले अप्रूवल लेंगे, but we will all go. So this is how our cell biology friendship continued. This is a very we are all from very, very different departments. We don't want anything from each other, but it's just that we are all together. We help each other, like someone said, that whenever it is required, just so you can imagine I am how much down the line as compared to you also now. So yeah, we'll be missing Rath a lot for this. And as I said that, I just forgot the content only. So as I said that Rath just permeates everywhere. I think there's a long way to go, and his pan ka stories go a long way. I think he knows that I like eating pan, so wherever if he goes somewhere, he'll get one pan of his kind and other sada pan for me, so that I have that pan. And I also remember in Srinagar, I think, Prabodh's meeting was to start just after our meeting began, and we all got together, and myself and Prabodh left at night. It was late at night, and because I wanted to join all of them the next morning at the airport, and they all went for a walk to have a pan or a cigarette, this thing, and the police came. And they were all in their Bermudas and chappals, because they just walked out. We had all walked out to leave. Aadhaar card kahan hai, ye kahan hai. They had nothing. So they were probably saved. And I know that many people thought that let's form a group like this. Such kind of a group should exist in NBRI. My husband was very envious of this group. कि ये क्या है, ये लोग यहाँ वहाँ वहाँ. So we didn't post pictures, we didn't tell him, because he formed a spouse group of which he was the administrator. He is the administrator even now. So Shrikant's wife, Prabhat's wife, BN's wife, everybody's wife is there, and Prabodh is the administrator of that group. And his job is to let them know what we are doing. So, Rath, we've had a very, very nice journey together, and keep that charm and swag in you. I think this is what makes you the Rath. And I am not going to say goodbyes to you at any cost. We are here in Lucknow. We'll age together, we'll die together. So, good luck, Rath.
Dr. Rath has a long time buddy in Dr. B.N. Singh. They did their master PhD together in CCMB, and then both of them eventually ended up working here in CDRI. I'll request him to come here and share some memories from older times.
Very good afternoon to all of you. इतनी सारी कुछ बातें कही जा चुकी हैं, but I think I will take you on 10 years before when he appeared in the CSIR, because I know him from his MSc days. And Alok was telling that ये रथ है कैसे खींचता है सब कुछ, तो don't forget that नाम के आगे श्रीकांत है. So Srikant himself is Vishnu, and then that his chariot, it's very easy for him to drive that. रथ की बहुत कुछ खास बातें हैं, बहुत हम राज हैं इसके, बहुत सारी बातें जो कहेंगे नहीं यहाँ पे. रितु ने बहुत कुछ पॉइंट आउट किया. We know that being seated here in CSIR, when I came here first, Rath was the only person I knew, someone I knew before from my PhD days, but she was in another campus. So हमें कुछ भी होता था तो हम भाग के रथ के पास ही जाते थे. ठीक दोनों हम लोग निकल जाते थे और मुंगा घर आए जुजुअल, जितना मर्जी एग्जेक्टिव जिसको सुनाना था सुनाते थे बनारसी अंदाज में. तो बनारस बहुत जिंदा था. हम लोग PhD टाइम में when I was in MSc, he joined PhD, and fortunately, unfortunately, he was the first PhD student of my own brother-in-law who was professor in the zoology department. So वो एक घर से भी जैसा जुड़ाव हो गया तुरंत इसके साथ. और MSc में से और PhD में हम लोग हॉस्टल में भी एक ही साथ रहते थे. स्पिरिचुअलिटी का जो पार्ट ये नर्चर करता है, मैनी ऑफ यू माइट नॉट बी नोइंग. गर्मिया हॉस्टल में अगर एक लड़का ऊपर सुबह सुबह देखता था सूर्य नमस्कार करते हुए, ये भाई साहब रथ जी होते थे. और ये उससे पूरी का पंडा है, ये जगन्नाथपुरी से आते हैं और टिपिकल पंडा है ये. तो वो वहाँ से स्ट्रेंथ खींचता है बेसिकली. मैनी टाइम्स वो हैज़ गॉट द अपॉर्चुनिटी टू स्टे विथ हिम इन द गेस्ट हाउस, एंड आई सपोज आई स्टेड विथ हिम हॉस्टल्स, इंसा गेस्ट हाउस एंड सो मेनी प्लेसेस. से बैंगलोर में भी जाते हैं हम लोग साथ में. ये भाई जी सुबह उठ के आधा घंटा पूजा करते हैं ये पूरा, एंड वेरी रिलीजियसली ही डू ऑल दिस थिंग्स. So I think somewhere at the corner he has that spirituality which drives him, and that how he manages to do such a diverse things which he do. His interpersonal skills are exemplary. I think I have not seen any people who can match that skill. And the kind of memory he has, जितने लोगों को याद करता है, आपको शायद आपके घर के बारे में जितना नहीं पता होगा रथ को पता होगा. और आप इसको किसी से मिलवा दीजिए, आपसे ज़्यादा दोस्ती उसकी उससे हो जाएगी. ट्रेमेंडस थिंग्स. एंड सामने हमारी शादी हुई इसकी CCMB में, हम लोग थे फ्लोर पे. वी यूज टू, गांधी वॉज देयर एट दैट टाइम, एंड इट वॉज वी यूज टू बी टोल्ड दैट गांधी से दिन में नहीं मिलना है. इफ़ यू वांट टू फेस गांधी, मीट हिम आफ्टर 6 PM. सुबह अगर पोस्ट प्री लंच में मिलेंगे तो मतलब वो बागी की तरह थे, आधी शक्ति उनके पास जाओ तो आपकी उनके पास चली जाती. तो आई थिंक रथ वॉज द ओनली पर्सन हु कुड डेयर टू काउंटर हिम इन द मॉर्निंग आवर्स. Though he was with Kitish Mazumdar, I was working directly with Ganji, but I never had this courage. गांधी रोज रात में आते थे, लाइब्रेरी से 4 जर्नल लेके आते थे. वो उनको ही वाज हैड स्पेशल प्रिविलेज टू गेटिंग नेचर, ये सब इशू नहीं होता था. ऐसी की CCMB की लाइब्रेरी जो है, गांधी यूज टू गेट रात में, ही हैज़ हैबिट ऑफ ऑल द स्टूडेंट जो इसके जो पेपर्स होते थे जिरोक्स करते थे, उनके टेबल पर रखाते थे. और वो सारे जर्नल्स हमारे टेबल पर रख देते थे कि सुबह 8 बजे के पहले लाइब्रेरी खोगे, उसके पहले वापस हो जाना है. सो आप रात में 2 बजे भी जाओ तो सुबह आके 8 बजे के पहले वो जर्नल वापस करना होता था. I had this, but I never had courage to talk to Ganji. Only Srikant could manage to talk to him. कुछ भी फ्लोर पे होगा तो भैया गांधी से बोलना है रथ को भेजो. इसका एक फ्रेज होता था, गिगुई पता नहीं याद है. He had a phrase which पूरा फ्लोर वही फ्रेज बोलता था. ये राम नाम की तरह जपते रहता था उसको वहाँ पे. And वहाँ से I remember when he came to CSIR and then my interview was there. That interview was postponed. I traveled to Lucknow without reservation and on came. I was sleeping in the hall. The main director office में interview चल रहा था. Abhay Sharma was there from the CMAT. So he had come to see me. भूपेन आया है शायद interview उसका. So in fact when they were announcing my name, I was sleeping. रात भर जर्नी किया था, बिना रिजर्वेशन का कुछ पता ही नहीं था. तो अभय दा केम एंड बोले कह तुम्हारा नाम बोल रहे हैं, जाओ भीतर तुम्हारा इंटरव्यू देंगे जाना. रथ वाज देयर, दे वर जस्ट सिटिंग बिहाइंड मी. interview happened and on. फिर इसके बाद वही as usual he telephoned me to my wife rather कि भूपेन काम हो गया है यार वो अब ये समझ नहीं आया कि कौन सा काम हो गया है यार तो फिर होल नाइट आई वाज रेस्टलेस यू नो दैट उस समय जॉब की तो ऐसी सिचुएशन होती थी कि होती ही नहीं थी एम्प्लॉयमेंट न्यूज़ हम लोग देखते रहते थे तो फिर रात में यानी फ़ोन कॉल मिला के हमने फ़ोन किया ईमेल में अजीब सा सिस्टम होता था उस समय बुकिंग आवर्स होती थी 1 घंटा उसमें ईमेल जाता आज करो तो सुबह पहुंचती थी दिस वाज अ सिस्टम तो रथ पे बात हुई तो ही टोल्ड मी दैट हम लोग थे रात में सीएम जी के साथ पार्टी थी तो इसने अमित को इंस्पिरेट किया अमित सिम्स आस्क सीएम जी कि क्या हुआ भूपेंद्र तो बोले नहीं हो गया उसको जा रहा है लेटर वगैरह जा रहा है ही एंजॉइंग सून so that's why he broke this news of me getting selected in CSIR-CDRI. Wife is की सविता whenever I visit his home the first thing she will tell कि भूपेंद्र भैया से बीएम भैया से इसको बोलिए कि घर में रहा करे so wonderful time Srikanth and I don't see you retiring. I myself is retiring next month so I think since 86 we are together and hope to continue this togetherness even after retirement. We are here in Lucknow. So all the best to you, Savita, Adit and Appu. They are also wonderful kids. Okay? And wish you a very happy post-retirement life. Thank you.
डॉक्टर रथ के फ्रेंड्स चारों पांचों इंस्टीट्यूट्स में भी हैं और उसके अलावा नॉन CSIR लैब्स में भी हैं इसीलिए डॉक्टर रथ को हम लोग पॉलीक्लोनल एंटीबॉडी कहते हैं ये किसी भी एंटीजन से इंटरैक्ट कर सकते हैं डॉक्टर पंत आई विल रिक्वेस्ट यू टू कम हियर यू हैव बीन अ लॉन्ग टाइम फ्रेंड मैंने आपको पहली बार उनके साथ ही देखा था
वेरी गुड मॉर्निंग सबसे पहले तो डॉक्टर मिश्रा एंड डॉक्टर रथ को सुपर एनिमेशन की बहुत बहुत शुभकामनाएं एंड ऑल साइंटिफिक थिंग्स हैव ऑलरेडी बीन सेड बाय मैनी पीपल हु आर एसोसिएटेड विद डॉक्टर रथ इन CSIR-CDRI. So basically I came in contact with Dr. Rath in 2002 in 10th 5-year plan में डॉक्टर रथ को एक डॉक्टर लालजी सिंह के साथ एक कोलैबोरेटिव प्रोजेक्ट था जिसमें SNP project tha and he has contributed in a lot in the association of SNP genes for the breast cancer in the diversified population of India and that has already been published in a good journal and later on he made a substantial contribution in the combinational drug where that cyclophosphamide along with the resveratrol to reduce the toxicity in the cancer patient. That is a big contribution of Dr. Rath and at the same time, if you say that in the country, the preclinical genotoxicity, that is known because of Dr. Rath, because he started this and expand the horizon of the genotoxicity, specifically in the regulatory framework, because that doing preclinical toxicity in bits and pieces here and there without any regulatory framework has no meaning in drug development. So he brought this concept to CSIR-CDRI that yes, we have to work together in a framework which is acceptable to CDSCO as well as to the US FDA and the EMA and other regulatory agencies. So that is immense contribution. And one very important contribution of Dr. Rath is that to bring the GLPs in CSIR-CDRI, because he came to me that no, and anyhow, because he was saying that Dr. Madhu Dixit is after me that no, GLP should be here. So for conducting or holding GLP in any government setup is a tough task because you have to deal with the starting from scratch to everyone, group 2, group 3 and scientists and everyone. So just make a team. So that means he led the team and made a vibrant GLP team here in CSIR-CDRI and I think 2 cycles have already been done. So over 6 years the GLP is there. 3 cycles, oh wonderful, and that on 11th the technical committee meeting is there. There your case is also there. CSIR-CDRI case is also there. The 3rd cycle is yet not completed, but don't worry because now this time the chairperson of the technical committee has changed and Dr. Madhu Dixit is a chairperson, next chairperson. So be sure that will be completed. No issue at all. So otherwise, beside the scientific part, so my association with Dr. Rath is immense, and as Dr. Ritu said very correctly, he can say anything, anywhere, to anyone. So that is his beauty. So बहुत सारी चीज़ें जो है नहीं बोली जा सकती, but he can do. So in brief, I'll say simply that all the very best for the future endeavors, and you are remain here in the Lucknow. So we'll definitely keep you in many of the committees in IITR as well, and definitely you will be in there in CDRI as well. So thank you so much.
There's a long list, but in the interest of time, we will have those discussions later. I will now request Dr. Rath to please come on stage and give his valedictory address and take us down the memory lane. Sir, please.
Still we are in morning session, and thank you so much to all my friends and director for इतने तारीफ मैं खुद रियलाइज कभी नहीं किया कि मैं इतनी अच्छा हूँ अगर इतनी अच्छा हूँ तो मैं क्यों लखनऊ में हूँ मोनिका तुरंत मेरे को काउंटर करने लगा है पुराना सिबलिंग मोनिका मी एंड बीएन वी आर सिबलिंग स्पीसीज फ्रॉम CCMB so always जीएन सिंह साहब को हृदय से धन्यवाद कि आप आए दो शब्द उनके बारे में मैं कहूँ जो भी आज शेड्यूल वाई का बात कर रहे हो शेड्यूल 2 का बात हो रही है न्यू NDCT रूल 19 में he is the person who initiated this as Alok bhai told एक दिन हम लोग को सुधीर बॉस ने फ्राइडे को बताया कि कल तुम लोगों को आना है यहाँ उस जमाने में हम लोग कैंटीन में CDRI कैंटीन में ताकि चाय नाश्ता की जुगाड़ पास में हो and Professor Dhawan, Professor P.K. Seth, Sudhir sir, and we are 2 attendants, toxicity attendants, Saras Sharma and me, and we actually learned the crafting of the Seedle Y. So that actually your initiative, sir, we could able to interact for the first time with Professor Dhawan and P.K. Seth very intensely, and I salute Dr. Sudhir sir, who actually encouraged us, could include us in the team, and we framed that Seedle Y. After that, of course, it is revised and some mistakes were incorporated. I told Dr. P.K. Seth how it can be corrected. He told, don't worry, we will take care of. So that's the first time I could know about him. As he told, 2004, that same meeting I was also there, for representative of CDRI. Manju Sharma was chairing the session, and they were so intense about implementing the developmental path, and many of CSR faculties were there, and we could learn that GLP and regulatory toxicity has to go together. If it has to go together, then it can go together only in CDRI. So if people are giving credit to me and Saras Sharma for GLP, I will not take that credit because the training was actually initiated by Dr. Sudhir. People went and get trained in outside several WHO meetings. Deborah Kiod was the person who used to be contacted to us, and they could able to find out Dr. Sudhir Srivastav. And Sudhir actually took lot of pain to become a WHO member, and almost every month he has to travel, not only in India but in Africa and West Indies, everywhere he traveled. And that is one of the reasons some of the colleagues became very, very envious of him. And ultimately, Sudhir was out of frustration, he actually left. He was in young age, he was to retire actually 2017, I remember his date of birth. But he has to, seven he actually left. Amit Bhai, who was always a pillar besides me, someone, as she told, for 5 years in the initial days, if somebody takes any of our name, they understand that these three will come always. Okay, that was our bonding. And during the launch time, actually, when we, after taking the launch, we move around from gate to animal house. So many people, they feel that perhaps they are the single unit. And that collaboration actually lasted till now. BN joined after 2000 August. After that, with BN, of course, Raj Kamal Tripathi and Ritu, we made a big group. And I think till this moment we are together. After retirement, I don't know Ritu will ask us or not, but I will. You see, cryptically she has taken that photograph. She is standing there, but she has instructed somebody perhaps to take that photograph. And I was sharing several of these photographs are with her. Morning, Prabodh sent me with Genda ke mala and other things. And that was in Khajuraho. And one, somewhere in Tamil Nadu, there was an election going, Kerala. And there was an election going on, and there was a leader poster, big poster next to our window of the Radisson Hotel. So I just saw that poster, and Ritu requested me, can you become like this? I immediately put the bed sheet and put that, right? And she took a photograph. I was not knowing that that photograph will be misused. All Tarif, yes, Sikandarabad, another accident, Sikandarabad railway station. I remember all these because somehow God has blessed me, as Bhupen told, the spiritual power is there. But the atheist, the greatest atheist in our institute, Amit Mishra, is the closest friend of me. And always we discuss several things, never fought till now. 27 years, 1 year over. Always, whenever there is a requirement, I just go down to Amit's lab, take a coffee or tea, then we discuss and come back. So that is the strength we have. I will talk about the students and my juniors. I never actually think that they are my juniors. Always, Amit, as yesterday he stressed, they are our peers. I am sure none of the young scientists or technical assistants can tell that Rasav के पास मैंने गया, मैंने प्यार से दो बात सुना नहीं. I encourage each and everybody to interact with the very closely with scientists, so that there is lot of barriers that will break. Actually, what happens, most of the people, they have some artificial barriers. मैं साइंटिस्ट जी हूँ, मैं क्यों बात करूँगा उससे? ऐसा नहीं है. घर में भी हम दादाजी से बात करते हैं और नौकर से भी बात करते हैं. We treat everybody as equal. So here also we should actually make a culture that we have to deal with, because these people, they are your strength, right? And if we are not dealing with them properly, how they will get back to you? So whether it is I start every day with this great smile of Purushottam Mishra. Anybody knows who is Purushottam Mishra? I am sure you are not knowing. I am telling who is that, and you will recognize now. Purushottam Mishra is the small man with very small hair, every day in front of the portico and security key where we are issuing, he used to clean, swipe, right? And whoever enters, he give a smile, if you remember correctly. And I know his family, his children, how he is dealing with that, how when he goes office, how he comes office and goes home, how he takes care of the children, how many daughters they have married and how many has to. I all have records with, because I talk, it takes only 5 minutes, but I share that pleasure, and he feels as if he spoke his heart to somebody else. So, Ganga Din. Anybody, anywhere, if they have some children somewhere, somebody is not getting admission or something, I asked Mujib to go to, drafted a letter and go to Rajnath Singh's office, spoke to his secretary that somehow you help this guy, his child has to be in the same Kendriya Vidyalaya, so that he can run the family with minimum of money. It is not a very big deal actually, it is only few minutes you can give, and they are all your only, right? After all, we are all Indians, we are all family members, we are all CSR family members. For our family also we are, if we are helping somebody, somebody will help our family. I am sure I have not given any very serious time to my children, neither son nor daughter. Always being told, ask him to stay in home, but I don't have that habit of staying in home. I also takes care of my own building where 28 families are there, and majority of them are actually seniors, and all their children are in USA and UK, well established. Every day evening they talk papa, mummy, but I have never seen, at least for the last 10 years, to come and see the so somehow I feel that I am, and they demand that. After 13 years of secretaryship, I told I will resign and somebody has to take care because I am also getting old. But all of them told no, the money has to stay with you. Whoever is president, secretary, we don't bother. All unanimously they selected me again, at least to, you will be relaxed as secretary, but you have to take care of the funds and its management. In New Hyderabad, it's a place for the last 21 years. I think majority of the family, they know Appu and me. Of course not her mother or brother, because we are the people every day going, talking, discussing. So that is what the nature I have built in, and all of I take care of, and I know majority of the students by name. As Ritu told, there is a name I always give, and sometimes Ritu also told me that don't speak like that to girls. They may take you nowadays. Rules are very different. Right. But Ritu, for your surprise, you will see many students are here. They come to me and tell, sir, your blessings required. Please pat my head today. There are many students there, right? They are not from only toxicology. So I think student own Rath as their teacher, as their mentor, and very transparently I tell. One incident I will tell. Sakil, correct. Swell's student, DAC member, she came, told, sir, DAC meeting is there, and I want to invite you. मैं कहा invitation क्या है, मेरा तो ये duty है, मैं जाऊँगा. Next meeting, some meeting shifting was there. So she went, मैं कहा तुमने अपने इच्छे से चेंज कर दिया, मैं अभी कैसे जाऊँगा, तुमको गोली मार दूँगा, तुम ऐसे गलती करते हो. सोहेल उसको बाद में बताया कि जाओ तुम सर को बुलाओ, तो कहा मैं ले जाऊंगा, सर बोले मेरे को गोली मार देंगे. So interactions has to be there, and it's nothing wrong. You are not losing anything. You have to interact with that, and that is what the quality perhaps I am building up. That's why I am very transparent everywhere, anywhere I can speak anything. That day in IGIV, director Maiti, when we were doing postdocs, Maiti and Ramanuj and Dr. Katti, who is Goa IT director, they were doing PhD. So they used to fondly call me Dada. So we were remembering the old days, how we are going Saturday, Sunday to Supravat to take our table, occupy in the first place so that we can sit for a long time. So things will not change for me. I am sure I will be active always. I will active in my own mohalla. I will active in CDRI. I will active in Lucknow. And there is a very big group, more than 50,000 people are there in Lucknow worldwide, internet worldwide, right? I am a member, and many of the members requested, sir, we want to give you farewell. What farewell? I am here only. No, sir, retiring farewell party हम करेंगे कहीं मलियाबाद में कहीं करेंगे. So I will be always connected. I am really grateful to each and every member of this CDRI. Each of you actually contributed to me, whatever habits or behavior or acumen or standards of research, everything I actually learned from all of you. Each of you is a teacher for me in one sense, and therefore I am grateful. Thank you so much.
Let's felicitate Dr. S.K. Rath now. May I request all the dignitaries, Professor Dhawan, Dr. G.N. Singh and Dr. A.B. Pant to please come forward, and our director, Dr. Radha Rangarajan, to felicitate him. I gather that Dr. Alok Dhawan has got some personal gift, some special gift for Dr. Rath. So to begin with, we'll start with sir. All of us, all of you. So there's a small token of respect and appreciation from all of us. So we are approaching the conclusion of this first session, and I now invite Dr. Amir Nazir to deliver vote of thanks.
Thanks, Smriti. Lots of emotions. It was one of the very rare moments to see at least Dr. Saman Habib get emotional, and then Dr. Rath as well. So, thanks everyone for all of these emotions, these memories, and we of course, all of us love Dr. Rath, and he has trained a generation of toxicologists, and he has trained all of us within the division, and we look forward to his guidance even after he leaves the institute. So lots of memories. I know there was less time. We had a long list of other people also who wanted to share. Some of our seniors could not join us. We have received their wishes. Dr. Madhu Dikshit sent her very warm wishes. Dr. Rath, she wanted to join, but something came up this morning and she could not join us. Our former head of the department, Dr. Neeraj Sinha, who is presently in Canada, he sent his warm wishes for you. And many others. There were our own technical staff from the division who also wanted to, but of course we'll continue interacting with them. And we'd like to thank our chief guest of the day, Dr. G.N. Singh. Sir, thank you so much. So we extended our invitation. He immediately accepted, and he said that CDRI is my own institution. How can I decline? So we greatly appreciate that warmth, sir. Thank you so much. And our very own Professor Alok Dhawan, who headed us very briefly and has been associated with not just CDRI, the entire CSIR family for very long. So he also immediately accepted our invites. Thank you so much, sir, for that as well. Dr. A.B. Pant, who has guided us as a GLP inspector whenever we require his help. For pre-inspection, we recently had an inspection and we requested him just a day before, and he came over and did a mock inspection and guided us, and he has always been there. And today also he accepted our invitation. Thank you so much. We'll be having a very insightful talk by him in the afternoon session. So thanks, everyone. So, of course, a big thank you to our own director, ma'am, Dr. Radha Rangarajan. So she, when I told her we are planning to organize this event, she immediately marked her calendar and just booked the slot. And she has been a guiding force throughout all of these preparations. Big thank you to you, ma'am. Thank you so much. And our former HODs who joined us, Dr. Sudhir Srivastava, Dr. C. Nath, Dr. R.K. Singh, and all our other colleagues who shared valuable memories with us here. A big thank you to all of you for being here. And thanks to Smriti for organizing this program beautifully. And in the end, we will invite all of you for a cup of tea outside. And please do join us for the session afterwards, where we will have 3 very insightful talks. Do join us for that. We will assemble here at 12 o'clock sharp. So it's 11:48. So by 12, please be back. And before we leave the hall, may I request all of you to rise and give a standing ovation to Dr. Rath. Thank you so much, everybody. We conclude this part of the program. Please be back at 12 sharp. Thank you.
I welcome you all back to the second part of the program. So we start with a talk by Dr. G.N. Singh. The title of his talk is Regulatory Tools for Medical Products and Clinical Results. So,
दे दीजिए और आगे पीछे कैसे करना है हमें किससे ये आगे ये क्या चीज एक बार दिखा दीजिए बस ठीक है एक बस ठीक थैंक यू So good afternoon ladies and gentlemen. Again I am standing in front of you. Just I am going to touch up some basic regulatory requirements. You know better than me, but I thought for the benefit of my young friends, I may touch up what exactly happening in Uttar Pradesh, in India, and what is the global requirements. What are the various regulatory tools available which you are aware, and I request you kindly go deeply so that all the regulatory requirements of the country, when we are known as the pharmacy of the world, when we are going to enter into the innovative phase, we may know the basic issues. So just I am touching what exactly is the. So as you are aware, these are the fundamental issues. The Drug and Cosmetic Act 1940. You see how lucky we are. Means our forefathers, they thought yes, India needs a very effective regulation because safety, quality, effectiveness, performance, these are the issues which requires a very strong network of the regulatory system. And 1940 onwards we are having such a wonderful arrangement. Afterwards, drug and cosmetic rules, you know, when act is passed, then for making the things to happen on the ground, some rules are to be made, and 1945 it was made. Afterwards a lot of changes has taken place, and we are fortunate that medical device rules, because few years back we were not aware much on the medical devices. Most of the things were treated as if it is the drugs, and it created a lot of hurdles for progression of the medical devices. This is how the stakeholders, the government and you people thought, let there be a very clear regulation, and in 2007 we passed on the medical devices rules. Though it is not the part of act, and the government is thinking seriously to have the Drug Cosmetic Act 1940 to be revised. We are going to have very shortly a very comprehensive and very effective act is to be put in place. The bill is already ready. It's due to some political or other reasons is lying here there, but I am quite confident that very shortly it would be coming. The new drugs and clinical trials 2019, that has become the game changer. I am just giving the outlines. When in 2009 India was a very progressive country in terms of conducting the clinical trials, global clinical trials, a lot of, तभी वो GLP भी आई, सारी चीज़ें आई कि कैसे हम प्रोग्रेशन को एड्रेस करेंगे. लेकिन हमारा सुप्रीम कोर्ट, हाई कोर्ट, जो हमारे सो कॉल्ड NGOs हैं, वो सारे लोग आके भारत को रोकना चाहे कि वी शुड गिनी पिग के उन्होंने चर्चा कर दी, सारा कुछ किया, जिससे कि हम थोड़े देर के लिए स्टॉप हो गए थे. जब भी हम US जाते थे, जब भी हम यूरोपियन कंट्रीज़ में जाते थे, तो लोग सोचते थे कि इंडिया में रेगुलेशन नहीं है. वो कहते थे कि दे आर क्लोज, जो रिलेशन बिटवीन रेगुलेटर एंड द प्रोड्यूसर एंड द अदर पीपल हु आर नॉट हैविंग द मीन्स एथिक्स टू मेंटेन द थिंग्स. तो ये हुआ कि नहीं, हमारा क्लिनिकल ट्रायल्स रूल्स इफेक्टिव आना चाहिए. 2019 में इसको पास कर दिया गया. इससे फ़ायदा ये हुआ कि नाउ द ग्लोब इज अंडरस्टैंडिंग कि इंडिया इज हैविंग ए वेरी स्ट्रांग रेगुलेटरी सिस्टम वेयर द सेफ्टी, एफिकेसी, क्वालिटी, द rights, well-being, and the overall safety issues of the subjects and the people are ensured. थोड़ा एक्ट के बारे में बता दें, क्योंकि जब हम लोग पढ़ते हैं तो सोचते हैं कि पूरा एक्ट बहुत बड़ी चीज़ होगी, लेकिन देखिए 1940 में बस थोड़े काम के लिए एक्ट बना गया था। जो ड्रग कॉस्मेटिक एक्ट कितने लोगों ने देखा है? नहीं सर, आप बच्चे लोगों से पूछ रहे हैं आप लोगों ने पढ़ा है कि नहीं एक्ट? देखिए हमारा एक निवेदन है, इसीलिए आज हम इसको टचअप कर रहे हैं कि द पीपल हु आर इन्वॉल्व इन इन्वेशन और द drug discovery, they must know about the regulation of the country and the globe. Otherwise, whatever the work we are doing, it is not going to meet going to see the day of life. इसीलिए हमारा आपसे निवेदन है, आप एक खरीद लीजिए। हम अपना बताते हैं कि जब हम पढ़ते थे VHU IIT में, तो इंडियन फार्माकोपिया वो तकिया कलाम मानी जाती थी। जैसे ही हम फर्स्ट ईयर में जाते थे, हमारे टीचर हमें खरीदवा देते थे और हम लेके रात दिन उसको सोते रहते थे, पढ़ते रहते थे। और उसका ये फ़ायदा हुआ कि हम एक एक चीज़ फार्माकोपिया का जानने लगे, तो हमारा एक्ट के बारे में जानने लगे। हमारा आपसे निवेदन है कि 350-400 वो भी पैसे की ज़रूरत नहीं, अब तो आप ऑनलाइन देखिए, सब कुछ अवेलेबल है। एक बार देख लीजिए, 20 पेज का केवल एक्ट है, देन यू वुड हैव ए फेयर अंडरस्टैंडिंग। क्योंकि हमने DCGI देखा हुआ है, जब आप बढ़िया काम करते हैं, सारा कुछ होता है, जब हमारे डायरेक्टर साहब लोग, आपके साइंटिस्ट जाते हैं रेगुलेटर के पास, तो क्रिटिसाइज करते हैं कि नहीं हमारी तो दवाई सबसे अच्छी हमने मॉलिक्यूल इन्वेंट किया है और आप पास क्यों नहीं करते हैं। तो सर ये हम आज बताना चाहते हैं कि हम लोग प्रो CSIR हैं, लेकिन आवर हैंड्स आर वेरी टाइट। वी हैव टू सी दैट वॉट एवर द रेगुलेटरी रिक्वायरमेंट्स आर देयर, दैट हैज़ टू मीट। तो आप लोग एक एक खरीद लेंगे, नहीं तो हम मैडम डायरेक्टर साहब से रिक्वेस्ट करेंगे कि आप ऑनलाइन इन लोगों को एक CD बनवा के दे दीजिए कि आप लोग एक बार पढ़ लीजिए, तो बड़ा आनंद आएगा। जो भी आप काम कर रहे हैं वो सब बिल्कुल। हाँ, तो देखिए उस समय जब बना था तो इम्पोर्ट, मैन्युफैक्चर, डिस्ट्रीब्यूशन एंड सेल्स ऑफ ड्रग्स एंड कॉस्मेटिक्स ही था। इसका नुकसान क्या हुआ? 1940 में मेडिकल डिवाइसेस का कोई मतलब नहीं था, मेडिकल टेक्नोलॉजी कुछ नहीं थी। आज तो ऐसा फ्यूजन हो गया है कि हमारी सारी चीज़ें वन दिखती हैं। अगर मेडिकल टेक्नोलॉजी में आप प्रांगण नहीं है, अगर आप मेडिकल डिवाइसेस में ठीक नहीं है, डायग्नोस्टिक में 2 पैथोलॉजी के लोग यहीं बैठे हुए हैं, देखिए बढ़िया रन कर रहे हैं आप, और ये क्योंकि इसका बड़ा स्कोप आने वाला है। मेडिकल डिवाइसेस इज गोइंग टू बिकम गेम चेंजर। डॉक्टर साहब जैसे प्रीक्लिनिकल स्टडीज, जैसे अभी हमारी बात हो रही है कि मेडिकल डिवाइसेस में कैसे वी कैन प्ले द राइटफुल रोल। वी वांट टू बिकम द फार्मेसी ऑफ मेडिकल टेक्नोलॉजी। अभी हम फार्मेसी ऑफ द मेडिसिन माने जाते हैं। अभी केवल आप समझिए कि हम 5% मेडिकल डिवाइसेस बनाते हैं, 95% गैप है। अभी सारी दवाइयाँ ख़त्म हो जाएंगी, सभी मेडिकल टेक्नोलॉजी में आर्टिफिशियल इंटेलिजेंस जो हमारी न्यू टेक्नोलॉजी है, दे आर गोइंग टू टेक ओवर एंड CDRI हैज़ टू प्ले द रोल विच इट प्लेड इन 1970, 80, 90s। जब भी हम लोग पढ़ते थे, देखते थे कोई भी ड्रग इन्वेंट हो रही है तो CDRI से होती थी। अगेन द कंट्री इज लुकिंग टुवर्ड CDRI एंड CSIR, हाउ वी आर गोइंग टू सी टू हैव ए डोमिनेटिंग रोल ऑन मेडिकल टेक्नोलॉजी। हमारी IIT कानपुर अच्छा कर रही है, IIT BHU कर रहा है, सभी लोगों के साथ हम लोग लिंकअप कर रहे हैं। हम केवल सिस्टाइज कर रहे हैं कि आप मेडिकल टेक्नोलॉजी में, जब मेडिकल डिवाइसेस में भी अपना फोकस करिए, क्योंकि हमने रूल बना दिया, सिस्टम इन प्लेस हो गया है। ये बकिया तो ये चीज़ें जो अभी हमने बताया वही है। मेडिकल डिवाइसेस रूल्स अब 2017 में आया, अब हमारे पास सिस्टम हो गया। क्यों हम लाए? जो अभी हमने बताया कि दवाइयों की तरह जब हमारे इंस्पेक्टर्स जाते थे, जो भी हमारे मैन्युफैक्चरर थे, इंपोर्टर्स थे, जब भी मेडिकल डिवाइसेस आती थी तो वो अपना दवाई की तरह उसको रेगुलेट करते थे। इससे ये हुआ कि इफ़ यू आर हैविंग ए टफ कंफ्यूज्ड रेगुलेशन, then the progress of the system, the science stops. And this is how we thought कि let the country have a very transparent, science-based and meeting the global requirements. अब हमारा सिस्टम इन प्लेस हो गया है, उसमें ये सारी चीज़ें दी गई हैं। अब हम आपसे इसीलिए कह रहे हैं कि जब तक कॉस्मेटिक एक्ट की किताब खरीदेगा तो उसमें रूल्स भी दिया हुआ है। आप सब जानेंगे कि भाई क्लास A क्या है, क्लास B क्या है, क्लास C क्या है, क्लास D क्या है। तो आपके अंदर बिल्कुल क्लियरिटी होगी कि जो एक्सपेरिमेंट हम कर रहे हैं, जो चीज़ बना रहे हैं, वो रिक्वायरमेंट्स को मीट कर रही है या नहीं। New drugs and clinical trials rules 2019, it has become a game changer. देखिए जब हम 2009 से 12 का सिनेरियो देखते हैं तो वी वर गिविंग क्लियरेंस टू मोर देन 120 new molecules were getting approval for introduction as a drugs in India. ये हम जब लोग रेगुलेटर थे तो इतना था. अब इसके बाद क्या हुआ कि पेटेंट रूल आया. पेटेंट के बाद जब प्रोडक्ट पेटेंट की आ गई तो हमारी जो बास्केट थी ड्रग्स की बिल्कुल कम हो गई. अब हम मुश्किल से 10-12 मॉलिक्यूल इंट्रोड्यूस कर रहे हैं क्योंकि शिफ्ट हो रहा है. बायोलॉजिक्स की तरफ हम जा रहे हैं और मेडिकल डिवाइसेस, मेडिकल टेक्नोलॉजी. IIT कानपुर देखिए वो मेडिकल टेक्नोलॉजी में जा रहा है. गंगवार उनका हॉस्पिटल खुल के तैयार हो गया है. सारे प्लेयर्स वहाँ जा रहे हैं. दुनिया देख रही है कि जितने हमारे स्टार्टअप्स हैं मोस्ट ऑफ देम आर टुवर्ड्स द मेडिकल टेक्नोलॉजी, मेडिकल डिवाइसेस, डायग्नोस्टिक. दुनिया बदल रही है. ऑर्गन, ये सारा चीज़ चेंज होने जा रहा है. दवाई की जरूरत ही नहीं पड़ेगी. हमारी आँख खराब हुई, आँख रिप्लेस हो जाएगी. बाल गायब हो गया, बाल आ जाएगा. आने वाला समय में ऐसा होने जा रहा है. तो हमारा ये कहने का मतलब कि आप लोग ब्रेन हैं, आप लोग स्ट्रेंथ है इस कंट्री के. हमारा आपसे निवेदन है कि इधर आप जरूर ध्यान दें. अब हम थोड़ा सा सीनरियो आपको बताएंगे. ये तो अपने ड्रग कॉस्मेटिक एक्ट रूल्स के बारे में बता दिए. अब आपको हम बता देंगे कि ग्लोबल सीनरियो क्या है, नेशनल सीनरियो क्या है, उत्तर प्रदेश कहाँ स्टैंड करता है. कि आपको बेसिक आइडिया होना चाहिए कि जहाँ पे हम बसे हुए हैं, जहाँ पे काम कर रहे हैं, हाउ वी आर गोइंग टू सी दैट इंडिया वुड बिकम ए वेरी प्रोग्रेसिव नेशन. यूपी इज गोइंग टू बी ए फेवरेबल डेस्टिनेशन इन टाइम्स टू कम. If I am giving this statement on behalf of the UP government, it means because the Honorable Chief Minister is working very hard on these lines, we are going to make UP to be a really different state. हम $1 trillion इकोनॉमी में हम लोग काम कर रहे हैं तो पहले हम भी कंफ्यूज थे कि $1 trillion इकोनॉमी हम कर पाएंगे कि नहीं कर पाएंगे. लेकिन आज हम दावे के साथ कह सकते हैं कि वी आर गोइंग टू अचीव इट. We are having very transparent system. We are all the department are working in just as a team spirit. उसमें CSIR के जो हमारे रोल हैं बहुत ही इफेक्टिव होने जा रहे हैं. हेल्थ केयर सेक्टर में हमारे धवन साहब बैठे हुए हैं. जब हम यहाँ आए थे तो हमने कहा कि देयर शुड बी ए लिंक बिटवीन CSIR and UP government. He pleaded and I am grateful to the DG CSIR कि she agreed. We had a MOU through which all the 36-37 labs are the partner to the UP government. We are going to we are taking their help. अभी कई चीज़ इनिशियल स्टेज में थी. हमारे जो CMAP है वहाँ पे हम लोगों ने एक ऑफिस भी इस्टेब्लिश की थी. किसी कारण बस उतना अच्छा भी नहीं चल पाया. लेकिन हम लोग ये करने जा रहे हैं कि ये सारी चीज़ें फंक्शनल होने जा रही हैं क्योंकि जब न्यू पड़ती है तो थोड़ा समय लगता है. हमारे कई सेक्टर्स बड़े प्रोग्रेसिव हो गए हैं. तो अब हम आपको दिखाते हैं कि ये ग्लोबल सिनेरियो देखिए. सब कुछ WHO, अभी आपने बताया कि हम WHO में जाते थे, श्रीवास्तव जी बता रहे हैं सब लोग. हम जाके WHO में रिप्रेजेंट करते हैं, हमारे रेगुलेटर जाते हैं, हमारे साइंटिस्ट जाते हैं कि दैट इज़ द न्यूट्रल एरिया फ्रॉम वेयर वी कैन टेल द ग्लोब कि नो इंडिया स्टैंड्स वेयर, व्हाट इज़ अवर विजन, हाउ वी आर गोइंग टू मेक इंडिया एज ए डिफरेंट नेशन. ग्लोबल हेल्थ सिनेरियो देखिए तो करेंट मार्केट जो है करीब करीब 12 ट्रिलियन की है. और अगर पूरा आप देखें कि विश्व की अर्थव्यवस्था क्या है तो करीब 90 से 100 ट्रिलियन डॉलर के आसपास की रहती है. उसमें देखिए कि इतना खर्चा करीब 12% हम हेल्थ केयर सेक्टर में करते हैं. तो एक बड़ा अमाउंट है, ये एक बड़ी व्यवस्था है. पैसे ही से दुनिया चलती है क्योंकि सारी अगर पैसा नहीं रहेगा तो सारी चीज़ें फोकस नहीं होंगी. प्रोजेक्टेड ग्रोथ जो है वो आने वाले 2028 में करीब करीब 16 ट्रिलियन डॉलर की हो जाएगी. कंपाउंड एनुअल ग्रोथ जो है 6.8 मतलब 7% के आसपास है, जो बहुत ही बढ़िया. अगर आप अदर से कंपेयर करिए तो वी स्टैंड मतलब इट इज़ ए फास्ट डिमांडिंग सेक्टर. क्या इसके की ग्रोथ ड्राइवर्स हैं? सब जगह दुनिया में देखिए एजिंग ग्लोबल पॉपुलेशन हो गई है, राइजिंग प्रिवेलेंस ऑफ क्रॉनिक डिजीजेज है, हेल्थ केयर पॉलिसी रिफॉर्म्स हर जगह आ रहे हैं. पहले अभी देखिए यहीं पे हमारी भारत सरकार ने जो हर व्यक्ति को अपना एक कार्ड दे रही है, उससे 5 लाख तक का फ्री ट्रीटमेंट पा रहे हैं. तो ये सारी टेक्नोलॉजी जो हमने कहा आर्टिफिशियल इंटेलिजेंस, रोबोटिक एंड डाटा मैटिक्स. अभी यही कल ही माननीय मुख्यमंत्री जी जाके डाटा सेंटर्स हमारे बहुत खुल रहे हैं Uttar Pradesh is going to be a leader in the country to have the best data centers, which would be collecting all the data, interpreting and making the science and the innovation on a faster mode. ये बहुत बड़ी चीज़ होने जा रही है, इसका फ़ायदा हम लोग को उठाने की ज़रूरत आ गई है। फार्मास्यूटिकल मार्केट कंट्री की देखिए तो करीब करीब हमारी जो ये ग्लोबल दिखा रहे हैं 1.48 ट्रिलियन है, मतलब करीब करीब समझिए कि 2% के आसपास जो ग्लोबल टोटल जीडीपी जो मानी जाएगी उसका 2%। तो अगर फार्मा सेक्टर बहुत बड़ी चीज़ है, आप एग्रीकल्चर का ले लीजिए, तेल का ले लीजिए, किसी और सेक्टर का ले लीजिए, तो दे स्टैंड नो वेयर। फार्मा सेक्टर मीन्स टेक्नोलॉजी मीन्स द फार्मा रिलेटेड थिंग्स आर मच हेड, जस्ट 3 times more than the other. प्रोजेक्टेड जो है ग्रोथ 2.1 ट्रिलियन, ग्रोथ रेट 5.7 है। और की मार्केट सेगमेंट क्या सबसे ज़्यादा प्रिस्क्रिप्शन ड्रग्स 60 to 70% है, जहाँ पे हम कहीं स्टैंड नहीं करते हैं। इंडिया में 2-4-10 मॉलिक्यूल लेट हमारे नित्यानंद साहब हमेशा बनवाते थे कि कितनी दवाइयाँ भारत में बनी हैं, उस वो हाँ 12-13 से कभी बढ़ी ही नहीं। जब भी वो बोलते थे जीएसटी जरा पता कर को बताओ कितना कोई बढ़ा, तो वो हम ढूंढ के देते थे कि नहीं सर इतनी है। वो बोलते थे अच्छा। देखिए क्या उनकी सोच थी। हमारा ये है कि अगर भारत को मजबूत बनना है, अगर वी वांट टू बी द लीडर इन हेल्थ केयर सेक्टर, then we have to be enter into the innovation, which Honorable Prime Minister always tell. Our Honorable Science Technology Minister, yesterday he was telling India has to enter into the innovative phase. अगर हम लोग इस समय नहीं करेंगे, और करेंगे क्या, हम लोग थोड़े लोग करेंगे, आप लोग करेंगे। जैसे जो हमारे रथ साहब ने बना दिया, श्रीवास्तव जी ने, मैडम ने जो डायरेक्शन दिया, जो हमारे धवन साहब ने बना दिया, हम लोगों ने तो बना दिया कि आपको एक टूल, एक प्लेटफॉर्म दे दिए हैं। नाउ यू पीपल हैव टू सी दैट हमारा प्लेन जो है हम रनवे बना के दे दिए हैं, अब आप उड़ाइए उसको बढ़िया से चलिए, हमारा निवेदन है। क्या ये देखिए जेनरिक ड्रग्स में तो वी आर द फार्मेसी ऑफ द वर्ल्ड। इट हैज़ गॉट, जैसे आप लोगों को मालूम है कि हम वॉल्यूम वाइज 3rd कंट्री एट द ग्लोबल लेवल हैं। वैल्यू देखिए तो 13 पे चले जाते हैं। अगर वही 3 हो जाए तो हम बिल्कुल डोमिनेटिंग हो जाएंगे। डोमिनेटिंग कब होंगे? जब हम इनोवेशन में जाएंगे, प्रिस्क्रिप्शन ड्रग्स में जाएंगे। ओवर द काउंटर बायोलॉजिक्स, बायोसिमिलर इज द फ्यूचर ऑफ दिस हेल्थ केयर सेक्टर, वेयर rightly the CDRI is focusing and other CSIR labs are doing well. We are very fortunate that they are focusing on that. ये तो हमारा आपको ये दिखा दें कि हम लोग दिखते कैसे हैं। अच्छा चलिए, दिखाना नहीं चाहिए। वो कह रहा है कि इसकी इकोसिस्टम फॉर इनोवेशन, यही हमारा सबसे फोकस्ड एरिया होगी, जिसपे हम चाहते हैं कि हम लोग विशेष ध्यान दें। वी हैव टू बी वेरी कॉशियस। हेल्थ केयर इकोसिस्टम देखिए, फार्मास्यूटिकल्स हो गया, मेडिकल डिवाइसेस हो गई, डायग्नोस्टिक हो गया, मेडिकल टूरिज्म का बहुत भारी स्कोप है इस देश में, बिकॉज वी आर गिविंग बेस्ट सर्विसेज, बेस्ट मेडिसिन, बेस्ट ट्रीटमेंट टू द पेशेंट। दिस इज हाउ मोस्ट ऑफ द अदर कंट्रीज जो अनडेवलप्ड हैं, डेवलप्ड के लोग भी आने लगे हैं, उनको हम देते हैं। तो इस मेडिकल टूरिज्म में जब हम हर ज़िले में एक अपना मेडिकल कॉलेज देने जा रहे हैं, बेस्ट हॉस्पिटल यूपी में देने जा रहे हैं, तो वी आर फोकसिंग दैट मेडिकल टूरिज्म वुड आल्सो बी वन ऑफ द इम्पॉर्टेंट कंपोनेंट ऑफ आवर जर्नी। मेडिकल एजुकेशन के बारे में बता ही दिए। नर्सिंग कॉलेजेस की कमी थी, अब हर जगह खुल गई हैं, लेकिन नर्सेज की जो क्वालिटी है, नाउ वी आर गोइंग टू फोकस दैट देयर क्वालिटी बी मैचिंग टू द रिक्वायरमेंट्स, to work out numbers, investment, technology, other things, hospital infrastructure। हमें एक कॉर्पोरेट कल्चर से निकालना है, क्योंकि यहाँ पे हमें भी खबर मिलती है कि जो बड़े हमारे जो हॉस्पिटल्स हैं वो सर्विस कम दे रहे हैं, पैसा ज़्यादा ले रहे हैं। तो ये गवर्नमेंट इज़ अवेयर, वी आर गोइंग टू डेवलप सब सिस्टम थ्रू टेक्नोलॉजी बेस्ड, हाउ वी आर गोइंग टू कंट्रोल, मॉनिटर दोज थिंग्स। इट वुड टेक सम टाइम, बट वी आर सीरियस ऑन दैट। जी सर, आप सही करें, 2 सेक्टर बड़े इंपॉर्टेंट हैं जो हम $1 ट्रिलियन में डिस्कस करते हैं। तो ये होता है कि आप दिखाइए कि हेल्थ सेक्टर में $1 ट्रिलियन में कैसे कंट्रीब्यूशन हो रहा है, और एजुकेशन सेक्टर में। तो हम लोगों ने ये कहा कि भैया ये 2 सेक्टर आप पैसे से न नापें, ये इसका नाप ये है कि जब अदर सेक्टर वुड बी परफॉर्मिंग वेल देन दिस सेक्टर वुड बी कंट्रीब्यूटिंग सिग्निफिकेंटली. दे आर द वेलफेयर सेक्टर. ये पैसा कमाने वाले सेक्टर नहीं है. अनफॉर्चुनेटली लोग जो घुस रहे हैं वो पैसा कमाने के चक्कर में घुस जा रहे हैं, लेकिन अब ये धीरे धीरे खत्म होगा. तो अब इसमें रेगुलेटरी ईज ऑफ डूइंग बिजनेस में इंडिया इज डूइंग वेल एंड यूपी इज पर्टिकुलरली एफडीआई. मतलब यूपी इज द फर्स्ट स्टेट विच हैज गॉट इट्स ओन एफडीआई पॉलिसी वेयर वी आर रिक्वेस्टिंग द पीपल टू कम इन्वेस्ट. डोमेस्टिक इन्वेस्टमेंट गवर्नमेंट पॉलिसीज. बकिया ये हम स्लाइड दे देंगे, आप लोग इसको थोड़ा जब खाली समय होगा तो पढ़ लीजिएगा. अब उत्तर प्रदेश के बारे में आ जाते हैं क्योंकि समय हमारा खत्म हो रहा है. क्या बता दीजिएगा? 5 मिनट. तो उत्तर प्रदेश फार्मास्यूटिकल मेडिकल डिवाइस पॉलिसी, ये हम लोगों ने एक चीज़ और दिखा दें कि क्या क्या चीज़ हमने किया है. ये तो कुछ गड़बड़ हो गया. कोई please bear with me just 1 minute. I want to show where exactly we stand. चलिए, उत्तर प्रदेश मेडिकल, we are having 4 components for Uttar Pradesh. One is to make the regulation and making ease of doing business. We brought a medical devices pharma policy. It is of unique in nature. UP is the first state which brought this policy after discussion with the stakeholders. Other states are following it. It has got a number of components which give fiscal incentives, regulatory incentives, science-based, other things are well incorporated. It was published in 2023, and again we are within a couple of days we are going to issue another government orders through which it is going to facilitate the growth, the going to hands holding of the pharma sector. So that policy incorporates most of the things. If you go on the web, you would be getting what exactly the pharma policy. ये 23-24 पेज का डॉक्यूमेंट है, इसको भी आप लोग पढ़ लीजिए. देखिए कि लोग पूछेंगे कि भाई हमें इंसेंटिव चाहिए उत्तर प्रदेश में, कैसे इन्वेस्ट कर सकते हैं, तो आपको ये सारा चीज़ जानकारी मिल जाएगी. मेगा फार्मा पार्क हम लोग ला रहे हैं. ये पार्क जो है हमारा ललितपुर में आ रहा है, क्योंकि इंडिया इज डिपेंडिंग ऑन API, key starting material, and the active pharmaceutical ingredients. The government decided to let India be self-reliant. That was the thought going on years altogether. Now 3 states are going to have centrally sponsored bug drug parks, and fortunately UP is going to have its own. और ललितपुर में बहुत ही अच्छा बन के तैयार हो रहा है. अभी हम पिछले सप्ताह में गए थे. 1500 एकड़ में हम लोग बना रहे हैं. सारी फैसिलिटीज, यू नेम एनीथिंग, वी हैव टू बीट द चाइना कंपटीशन. सो वी थॉट कि ऑल द कंपोनेंट्स विच रिक्वायर्स टू बी पुट इन प्लेस इज देयर. आप लोग मैडम, हाँ, आपसे निवेदन करेंगे कभी एक टीम लेके चलिए. यू सी इट एंड हाउ CDRI कैन प्ले द राइटफुल रोल ओवर देयर. क्योंकि हम एक मैसेज देना चाहते हैं कि हमारे पास ताकत है, हमारे पास साइंटिस्ट हैं, हमारे पास सिस्टम है. अगर आप आएंगे, कोई भी API में फर्दर इम्प्रूव करना चाहते हैं, की स्टार्टिंग मटेरियल में कोई हेल्प लेना चाहते हैं, तो माई बेस्ट इंस्टीट्यूशन इज़ अवेलेबल ओवर हियर. और आप 3-4 साइंटिस्टों को एक बार भेज दीजिए. जब हम जाते हैं तो ले आके इनको दिखाएंगे. सीइंग इज़ बिलीविंग. जब आप लोग देख लेंगे कि भाई क्या बन रहा है, अभी हम बोल रहे हैं तो आपको लगता होगा कि ऐसे ही डॉक्टर साहब हवाई बात कर रहे हैं, लेकिन इट इज़ रियलिटी इज चेंजिंग. ये सब चीज़ें बन के तैयार हो गई हैं. करीब करीब हमारी जो जितनी कॉमन फैसिलिटी, जो भी आप नाम लीजिए, हम क्यों पीछे हो गए थे? क्योंकि चाइना हमारी, देखिए IDPL, हम IDPL के एम्प्लॉई रहे हुए हैं. IDPL वाज द मदर यूनिट फॉर ऑल द बग ड्रग्स फॉर्मुलेशंस, chemicals, or you take anything. And why we could not able to survive, it is due to stiff competition from China. They were making in bulk. अगर हम पेंजी 50 मतलब 1000 में बनाते थे तो वो 5 रुपये में बना के देने लगे, और जब हम बंद कर दिए तो वो 1 लाख में देने लगे. दिस इज द एक्सप्लॉयटेशन टुक प्लेस, एंड दिस इज हाउ वी आर आउट ऑफ द पिक्चर इन एंटीबायोटिक्स, antimicrobial. So we are seeing that in times to come we have to become self-reliant, so that इंडिया कीप ऑन प्लेइंग द रोल ऑफ फार्मेसी ऑफ द वर्ल्ड। तो उसके लिए सारी चीज़ें की ज़रूरत है जो हमारे पास तैयार हो गई है। मेडिकल डिवाइसेस पार्क ये एक बहुत ही अच्छी चीज़ हो रही है। इट इज़ ऑफ यूनिक इन नेचर। ये जो जेवर एयरपोर्ट है उसके ठीक बगल में जब हम लोग आए तो माननीय मुख्यमंत्री से निवेदन किया गया कि इफ़ इंडिया वांट्स यूपी वांट्स टू बी नंबर 1 देन वी हैव टू प्रोवाइड द बेस्ट फैसिलिटीज़। फॉर्चुनेटली जेवर एयरपोर्ट के पास जो सब एकदम बगल में जो ज़मीन थी माननीय मुख्यमंत्री जी ने 350 एकड़ इस पार्क के लिए दे दिया है। हमारी करीब करीब 90 यूनिट्स वहाँ पे ऑलरेडी कंस्ट्रक्शन में आ गई हैं, कॉमन फैसिलिटीज़ इन प्लेस हो गई हैं। आप चल के एक देखिए, कभी भी आप जाइए तो बताइए, हम वहाँ के लोगों को बता देंगे। एकदम जेवर एयरपोर्ट के पास ही सड़क के से आप जाएंगे एक्सप्रेस वे से तो दाहिने तरफ इधर से जाते समय पड़ता है। गो एंड सी हाउ द यूपी इज़ चेंजिंग। तो आपको लगेगा कि नहीं, अगर कोई अपनी यूनिट लगाना चाहता है तो लगाने में उसको सुविधा हो जाएगी। ये कंपोनेंट्स ऑफ द पार्क हैं वो हमारी स्लाइड से मिल जाएगा। ये सारी चीज़ों के लिए हम लोगों ने सोचा कि भाई कैसे सफल होंगे, बिकॉज लॉन्ग एक्सपीरियंस ऑफ माइन इन इंडस्ट्री, इन रेगुलेशन, इन स्टैंडर्ड सेटिंग इंस्टीट्यूशन, व्हाट वी वर लैकिंग इज नेटवर्किंग। हर आदमी जैसे CDRI भी बन गई, CMAP भी बन गया, IIT भी बन गई, सब कुछ बना, लेकिन लिंक नेटवर्किंग मिसिंग हमें लगती है कि हम अपने टेबल पे तो सबसे धनी आदमी हैं, लेकिन जब बाज़ार में जाते हैं तो हम अपना धन छुपा लेते हैं, किसी को दिखाते नहीं कि यह तो सब कुछ यूज नहीं होता है। 15-20 साल के बाद वो पूरा हो जाता है। तो नाउ द टाइम हैज़ कम व्हेन वी मे एस्टैब्लिश स्ट्रांग नेटवर्किंग। देन वी डिसाइडेड एंड वी आर वेरी फॉर्चुनेट, मैडम हैज़ आल्सो कंट्रीब्यूटेड इन many of the discussion. We thought ki how we can build up an institution for innovation and research. तो हम लोग पहले यूनिवर्सिटी बनाना चाहते हैं, फार्मास्यूटिकल यूनिवर्सिटी। माननीय मुख्यमंत्री, बस ये लास्ट स्लाइड है, हम लोग गए तो उनसे कहे कि एक फार्मास्यूटिकल यूनिवर्सिटी बना दी जाए, जिससे कि जो 400 हमारी इंस्टीट्यूशन है, क्वालिटी ऑफ एजुकेशन सबसे ख़राब हो गई है, उसको हम लोग रेस करें। तो उन्होंने बोला कि नहीं, अभी इस काम ये तो अभी AKTU के साथ रहने दीजिए, आप लोग एक इंस्टीट्यूट बनाइए। तो हम लोगों ने सोचा कि हमारे इंस्टीट्यूट तो इतने बढ़िया बढ़िया बने हुए हैं कि इंस्टीट्यूट रिसर्च डेवलपमेंट के लिए बनाने का कोई मतलब नहीं है। तो उन्होंने बोला कि नहीं, इसको आप बायरैक की तरह सेक्शन 8 कंपनी बनाइए। तो हम लोगों ने एक सेक्शन 8 कंपनी बनाई। इसका केवल काम ये होगा कि नेटवर्क करेगा जो हमारी इंडस्ट्री से रिलेटेड इशूज़ हैं, जो हमारी एकेडमिक इंस्टीट्यूशंस के इशू हैं, जैसे स्टार्टअप्स हैं, उनके क्वालिटी कैसे हम रेस करेंगे, उनके लिए इनोवेशन में क्या क्या ग्रांट भी देंगे। जैसे मान लीजिए अभी तो नहीं, लेकिन आने वाले 3-4 साल के अंदर अंदर हम लोग ग्रांट भी देने लगेंगे। जैसे बायरैक देता है, कोई अच्छी रिसर्च हो रही, कोई अच्छी चीज़ हो रही है तो उसमें फैसिलिटेट करेंगे। CDRI में सब लोग नहीं आ सकते, लेकिन प्रमोट फार्मा के थ्रू जो हमारे इंडस्ट्रियल हाउसेस 500 इंडस्ट्री आ रही है उत्तर प्रदेश में, तो उनकी इशूज़ होंगे। उस इशूज़ को एड्रेस करने के लिए एक फैसिलिटेटिंग इंस्टीट्यूशन चाहिए। तो उसके लिए ये प्रमोट फार्मा बनाया गया है। और इसकी आज ही मीटिंग 4 बजे है, बोर्ड ऑफ डायरेक्टर्स की है। हम लोग जाकर उसमें सारा चीज़ लीड करेंगे। हम इसके बारे में भी बताएंगे CDRI के साइंटिस्ट लोग और कैसे इसको देखना चाहते हैं। धवन साहब इसके बारे में जानकारी थोड़ा रखते हैं। तो ये मान के चलिए कि इंडिया इज़ चेंजिंग। और ये बायोटेक पार्क में हम लोग अभी शुरुआत करने जा रहे हैं। वहाँ पे उसको इस्टेब्लिश मतलब एक बिल्डिंग हमने ले लिया है। तो उसमें बहुत छोटा ये इंस्टीट्यूशन होगा, लेकिन इट इज़ लुकिंग to have a very close and effective coordination with industry, academic institution, including the CSIR labs. ये सब कैसे हम कर उसके पॉलिसी लेवल पे, गवर्नमेंट को पॉलिसी लेवल में जब कोई इनपुट्स चाहिए तो ब्यूरोक्रेट लोगों को तो पता रहता नहीं। हर आदमी वहाँ काबिल बना रहता है कि नहीं, हम ही जानते हैं जी, हम ही जानते हैं, वो किसी को सुनते नहीं है। तो उन्होंने सोचा कि पॉलिसी लेवल पे इंटरवेंशन करने के लिए इंस्टीट्यूशनलाइज मैकेनिज्म होना चाहिए। तो अब ये प्रमोट फार्मा जो होगा वो इंटरेक्ट करेगा। अगर कोई फार्मास्यूटिकल पॉलिसी बनानी है, हमें कोई ग्रांट देनी है, किस एरिया में रिसर्च होना है, तो इट वुड बी फैसिलिटेटिंग। और ये इसका सिस्टम है गवर्निंग बॉडी का। इसमें 3 हम लोगों ने इंडिपेंडेंट डायरेक्टर्स रखा। और आई आल्सो हैपेंस, पहले हम नहीं बनना चाहते थे, हमने कहा कि डायरेक्टर बन के क्या करेंगे। फिर गवर्नमेंट ने डिसाइड किया कि नहीं रहेंगे तो मिस हो जाएगा, जिसे कंसेप्चुलाइज जो मतलब इंस्टीट्यूशन बन रहा है। तो देन आई एक्सेप्टेड एंड टुडे वी आर गोइंग टू हैव द बोर्ड मीटिंग ऑफ द प्रमोट फार्मा। वेज फॉरवर्ड द केस स्टडी ऑफ UP, आई एम शोइंग हाउ द the best other state would be following how the scientists are going to make that India become an innovative nation. Fusion of healthcare with current medical innovative tools would bring safer, effective, quality medical products and services to the user. हमारा ये निवेदन है, पुनः हम आपसे कह रहे हैं कि यूपी is changing state. We are going to see that it enters into the right phase, and we people are very fortunate that we have got birth at a time when India is going to become an innovative nation, where we are going to have विश्व गुरु बनने की इच्छा रखते हैं और बनेंगे भी. ये बात आप मान के चलिए, कभी मन में कन्फ्यूजन मत लाइएगा कि ये जो स्टेटमेंट माननीय प्रधानमंत्री जी दे रहे हैं, हमारे डायरेक्टर साहब कह रहे हैं, हमारे मंत्री जी कह रहे हैं या हम लोग कह रहे हैं, क्योंकि जीवन का हमने देखा है. आपके सामने हम खड़े होकर अगर स्टेटमेंट दे रहे हैं तो आप ये मान के चलिए, जब हम पढ़ रहे थे तो हमें लगता था कि नौकरी नहीं मिलेगी. जैसे आपने कहा कि एम्प्लॉयमेंट न्यूज़ हम पढ़ते थे, हम जाके रुइया एक हॉस्टल था हमारे यहाँ, वहीं बैठ के हम लोग वही देखते थे कि हमने एम.फार्म कर लिया, पीएचडी किया, नौकरी तो मिलेगी नहीं. लेकिन इसके बाद देखिए कि वी आर हैविंग ए डोमिनेटिंग रोल एट द ग्लोबल लेवल. अब हम लोग ये ऐसा है कि जहाँ जाते हैं, जैसे WHO में हो, US में हो, किसी कंट्री में जाइए तो जब तक इंडियन साइंटिस्ट रेगुलेटर्स आप लोग नहीं रहते हैं, उनकी मीटिंग नहीं होती है. पहले वो कभी केयर नहीं करते थे, कभी हमें नमस्ते नहीं लेते थे. आज अगर इंडिया का रिप्रेजेंटेशन नहीं है तो वेट करते हैं कि नहीं हमारे रथ साहब कहाँ गायब हो गए. बोलो, हमने ऐसा अनुभव किया है. पहले हमने देखा है, इसलिए बता रहे हैं कि आप सर हमने ये अनुभव किया. पहले हम जब UK में पढ़ते थे तो बस में बैठते थे, लोग किनारे हट जाते थे. आज जब हम उसी लिफ्ट पे जाते हैं तो गुड मॉर्निंग, गुड इवनिंग वो अंग्रेज करते हैं, तो हमें लगता है कि नहीं हम वही ब्लैक हैं, लेकिन हमारी ताकत बड़ी है ड्यू टू माई परफॉर्मेंस. आज हमारी यूनाइटेड स्टेट फार्माकोपिया आप लोग देखे होंगे, नित्यानंद साहब ने जब इंडियन फार्माकोपिया बनाया तो लगता था रोजर विलियम साहब उस समय उसके CEO थे, वो समझते थे कि हम बड़े डोमिनेटिंग हैं. जब हमने इंडियन फार्माकोपिया बनाया, नित्यानंद साहब ने गाइड किया, तो आज वो हमारी जो स्टैंडर्ड्स हैं वो अडाप्ट करते हैं. हमने MU किया, हम उनको देते हैं, पहले उनसे लेते थे, आज हम देते हैं. तो ये स्ट्रेंथ है CDRI की ये स्ट्रेंथ है. सेंट्रल कॉमन डॉक्टर साहब हमेशा अभी 1 साल पहले तक जब भी हम जाते थे, वो एक तरह से जैसे मान लीजिए हम दोनों लोग गए थे तो हाथ पकड़ के वो यही कहते थे, GN सिंह ये बिल्कुल सेफ ड्रग है, इस सेफ्टी का तो कोई इशू नहीं है, इसमें इतने इसके अभी थेरापेटिक यूज़ हैं, एंटी कैंसर एक्टिविटी जो हम देखना चाहते हैं कि इट कैन बी ए गेम चेंजर. इस पैशन से उन्होंने काम किया है. और उस तरह के कई मालिकूल आपके हमने एज ए DCGI देखा है. IDPL में जब हम थे तब यहाँ से IDPS के कई आपकी दवाइयाँ हमारे यहाँ टॉक्सिसिटी में टेस्ट होती थी. हम ये चाहते हैं कि अगेन यूपी में जो भी ड्रग डिस्कवरी, रिसर्च, इनोवेशन और टेक्नोलॉजी ट्रांसफ़र होने जा रहा है, CDRI में वुड बी प्लेइंग द राइटफुल रोल. स्टार्टिंग ओनली दी IIT से लोग आए थे, वो करीब करीब 300 से 400 बिलियन US$ की एक क्लिनिकल ट्रायल करने जा रहे हैं. आई रिक्वेस्ट ऑल ऑफ यू टू ज्वाइन हैंड्स. वी हैव टू इंटर विद द इंडस्ट्रियल पार्टनरशिप so that the people who are going in different countries, they may do the preclinical studies in our GLP certified lab. With the help of the scientists, we would be succeeding. With these words, I express my gratefulness to all the people, especially the director Najir साहब, और हमारे मिश्रा जी तो अभी दिख नहीं रहे हैं, तो उनको भी हम धन्यवाद देते हैं. मैडम, आई एम ग्रेटफुल to you for giving me an opportunity to speak in front of such a vibrant, dynamic and future of the country. Thank you very much.
Thank you, sir, for your valuable insights and such an inspirational talk. We move to the next talk of the session, which will be delivered by our chief guest, Professor Dr. Alok Dhawan. The title of the talk is CSIR Supporting Risk in Science, Lab to Policy. Sir.
Thank you, Smriti. And let me at the outset thank Director CDRI for giving me this opportunity. Perhaps I'm the only one who can claim that the DNA in me belongs to CSIR, because my dad was in CSIR, and so the only very passionate. I mean, I was very sort of moved when I got this invitation from Amir, and I couldn't say no, of course, coming to CDRI. And that's where my childhood was. And to have headed a CSR lab, so on the one side was pharmacology, my dad, and I was on the toxicology side. But nonetheless, we had a good relationship at home. But again, the only father-son duo who ever headed a CSR laboratory. So I think it only goes to show how CSIR has nurtured risk, not only with personnel but also with the projects or things that were challenging or perceived to be challenging at one time, but CSIR stood firm with its scientists, and that is why CSIR is where it is today. And I think we, as Dr. G.N. Singh very rightly mentioned, we have the future of the country in front of us. They should take the leap of faith, they should take challenges, and I'm 100% sure that the present dispensation will also support that risk. As is said in every sphere, I think you don't put all your eggs in one basket. So you should have, and that's what I followed when I was heading CSIR, IITR. Some part of the process, the money should be put in risk-taking projects which are supported. I will perhaps share a story of my own as far as the risk-taking ability is concerned and how it was supported. But I think it is, if you look at CSR as a whole, the institutions, especially when Radha would appreciate when you go to directors' conferences and you see what is happening, it is unbelievable, right? I mean, there are directors who are projecting 6,000 crores over a period of 5 years, and they want to bring out aircrafts, and they've actually come out with it. And the fact that the government has supported it. So I think unless we are able to take that risk, small risks, but of course you have to take that leap of faith. So few examples. One, before I get into the, this is the run of the mill, so but I'll share anecdotes with you that are more important, I think. And how it leads to policy. G.N. Singh gave a wonderful example. Unless you get good quality science ahead of time, you don't get policy of your own. You are a follower. I think Dr. Mashelkar recently mentioned very well that you have to come up with next practices. You can't be following best practices. So I think that is where the difference is between a leader and a follower. So if you are a leader, you come up with next practices. If you're a follower, you keep following the best practice available in the world. So I think that's the difference, and that is where I feel CSIR can do wonders. And I still remember when one of these 10th 5-year plans we were writing, and I wrote as a scientist that I want to start in silico toxicology in the institute, and the director called me. आप जानते हैं क्या होता है? मैंने कहा नहीं जानता हूँ सर. तो ये कौन करेगा? मैंने कहा मैं करूँगा. तो आप कैसे करेंगे? मैं सीखूँगा सर. तो मैं आपका नाम इस पे लिख देता हूँ. मैंने कहा लिख दीजिए सर. बाकी सब ने वही लिखा था जो कर रहे थे. थोड़ा सा आगे बढ़ाएंगे तो प्रोजेक्ट मिल ही जाएगा. Ultimately, it turned out that it was sanctioned in the 5-year plan. Now, then I was, of course, in a soup. I mean, how do you begin? But then we caught hold of a company who was indigenous in the country, and they were into drug development. But they wanted to do toxicology, in silico toxicology, and they said, why don't we join hands? So they sort of were agreeable that they can modify the software for toxicology, and that's how we began. And currently, our institute, IITR, has the high performance computing facility for in silico toxicology. So where it started to where we are now, I think is phenomenal. I think small things that you can do to ensure that we are at the cutting edge of science technology in whichever field that you're working. It's always good to jump the field. It's much better, to be honest. I changed my life 5 times in 25 years, but thoroughly enjoyed. And I must say that I was supported all along. So I will just share, I think I will move on from here. That's the current institute where I am now. That's the Center of Biomedical Research. Again, something that is very unique that state government is supporting such an institution. It is unbelievable the amount of infrastructure and the amount of investment that we have from the government and continuously being supported. What I would be sharing with you is perhaps what I've done at CSIR. I thought that will give you a glimpse of what is achievable, what can be done. And some of the initial slides were when I think about 8-10 years ago I was talking to young scientists in HRDG, and that's where I thought that's the routine Council of Scientific and Industrial Research, but actually this is what it is, right? So I think if we understand that CSIR is there to foster innovation, CSIR is there to foster innovative research, I think that is how you begin. That's the starting step for students, for scientists, for directors as well. Once you take a step forward, I think people are there to believe in you. And that is why I think over the period of time, they have now changed the logo to the Innovation Engine of India. And I think I'm very proud of it that end of the day, the government also realizes that if in any field they want to move forward, they have to come out to CSIR one way or the other. You look at any big missions of Government of India, CSIR is always part of it. It is not possible that CSIR can be ignored at any point in time. So I think we should be proud of it. The fact that majority of the policies related to various fields, I mean, especially the youngsters, they're perhaps not aware. The building code of this country belongs to CSIR. The road, how they're going to be prepared, is belonging to CSIR. So it's all the science that CSIR has done based on the funding and based on the sort of understanding with the government that what we are doing will be helpful. So we don't have to borrow things. And I think one thing I'll just like to mention here, we are among the first in terms of science technology. But the interesting part is that as things move forward, CSIR was always intertwined with each of these institutions. And even today, if you look at these institutions, they always ask to follow the systems that are placed in the CSIR system. So I think there are a lot of things that these organizations have borrowed from CSIR. And so therefore I think it's very important and pertinent that we keep evolving, but at the same time not lose track of what we stand for as CSIR and how we have been supported over the decades that CSIR has been in existence. And as Dr. G.N. Singh sahab rightly mentioned, that is where we are headed to, sir. Right? पहले हमारी डिपेंडेंस थी, अब हम इंडिपेंडेंट हैं और मैं बहुत शुक्रगुजार हूँ जी.एन. सिंह साहब का, इन्होंने यूपी में आने के बाद एकदम काया पलट करी है, क्योंकि वो यूपी को समझते हैं और इंडस्ट्री को समझते हैं और यूपी गवर्नमेंट इनके ऊपर भरोसा करती है. उन्होंने बहुत बड़ा रिस्क लिया इनको लाके, लेकिन वो रिस्क फ्रूटफुल हुआ. यही मैं कहना चाहता हूँ. देखिए हर चीज़ में रिस्क है, कैलकुलेटेड रिस्क लेना एक चीज़ है, बट इफ़ यू वांट डिसरप्शन तो आपको रिस्क एट एनी कॉस्ट सपोर्ट करना है. It may not be 100% risk support, but it could be 50%. You're playing safe, and 50% you're supporting risk. 10% you're supporting disruption. There are several examples, and I can go on and on with examples where disruption has happened because they were supporting the risk. But we have to stick to the title, which is policy, right? And if you do good quality science, that is how policy comes through. And it is very important that, again, I'll say that CSIR institutions have played a stellar role in developing policies across the board for the government of India. All right, it could be food, it could be pharma sector, whatever sector you name, and the policies are sort of coming out of the science done at CSIR. And that is why they are there. So I'll just share with you some of the examples, a story rather, how it started and Rath तुम्हारी जैसी photo है नहीं, बचपन की photo है. This is 2005, okay. Me standing, and this is actually the photograph of my house on that day. It was as cold in Michigan. I was invited for a meeting. I had gone there to do some other work, and I was invited for a meeting by some chemical engineers on the Friday, and they said, we'd like to discuss with you about some products that we made, and we want you to do the toxicity when you go back home. The discussion was that we have made fullerenes, and we want you to do the toxicity. I had no clue what fullerene was, to be honest. It is 2005. But then you have to act smart. So I think CSR teaches you a lot of things. So I said the first thing, I'm a federal government employee. I can't commit anything between India and the US. So how about I come back to you by next week? Oh, he said absolutely fine. The fullerenes are with us and there's no issue. So first thing, I went back and read about what is nanomaterial, what is a fullerene, what are colloids, etc. The second thing I had to do was to, of course, as they say, the boss is the most powerful. In our case, the lady sitting in front. Without the director, nothing moves. They have to support the rest. So this is what I wrote, and this is where I come to leadership. This is what I wrote to CM Gupta, who was the then acting director of IITR or joint charge of IITR. Is that email that I wrote to him, actual email that I wrote to him, and I thought that we should start the nanomaterial toxicology in this country, in India. And this is what he wrote: Dear Alok, please go ahead, best wishes. That's about it. And when I came back in 2006, he said, "Tumhe chahiye kya ye batao?" And then we just started within no time. Everything was in place. This was that paper that I'm telling you. For the two individuals, Syed Hasham and Vladimir, these two guys from Michigan State, for them this is the most cited paper in their career, right? For us it was the best thing because we got a well characterized starting material to begin with, right? We just had to do the toxicity of this material, everything else was characterized by them. So this was the starting point where we thought, and we still get citations for this paper because it was an interdisciplinary thing that we did. Okay, I will now, what time do I have to finish? वो बता दो यार, तो रथ का टाइम वेस्ट नहीं करना है. 105, wonderful. So I'll skip some slides and then I'll come to where the problem lies. So you'll appreciate how, okay. So these were some issues that were faced when we started because we did not know if the fullerenes were coated with any, let's say DMSO or dimethylformamide or anything that they were being prepared with. So if that goes along with it, then it causes toxicity, not the fullerene. The solvent might cause toxicity. So these were issues that we had to deal with. There were other issues that as I move, perhaps you'll have a sense of where we were going. But everything kept documenting. We kept documenting everything that we were doing, and so those days there were two compounds that were being used extensively. One was zinc oxide in the cosmetics, and titanium dioxide in cosmetics and pharmaceuticals as well. So we thought, let's go with zinc oxide to begin with and give you a storyline. And we used, of course, whatever we used as the cell line to begin with, and did the in vivo experiment. Don't worry about all that. And each of these slides I'm telling you are now the sort of Bible for others to work. That much I can assure you. The way things were done that time, and a lot of collaboration with CCMB for electron microscopy. We didn't have one, and unfortunately CDRI was very bureaucratic to work with. You wouldn't imagine. Anyway, अब तो कह सकते हैं ना. I don't know if it has now become more liberal. I don't know, but very difficult to work on an electron microscope. कहाँ है हमारे electron microscopy वाले कहाँ हैं? नहीं है. बाजपेयी जी थे. Anyway. Correct, but put to no use, right? Let's be honest. Yeah. That's how things change. That's what I'm saying. But anyway, a lot of help, lot of fundamental science behind it. But why I'm telling you all this is how much was at stake for me, being at 40 in terms of age, had no clue what nanomaterial is. Entire world was working on these particles. Everybody had their own point of view. You had to sort of push every single paper through the Europeans and the US, and they would say, no, this is all crap. Then you have to write back again and write to the editor and so on and so forth. It was a Herculean task. For the students, I will say, trust your guide, not blindly, but of course trust him or her. And I would give this away to my students who trusted me कि यार ये क्या चीज़ करवा रहे हैं, मुझको कुछ हवा नहीं है, मतलब what. Of course, everything was very new in India. Just to give you a sense, when the nano mission program came to us, the first review was in IIT Delhi, So I just went through the nano mission. There were 200 projects sanctioned. 199 were on synthesis, 1 on toxicology, and that was me. That's it. So for some reason the students thought, ki chalo kuch to kar raha hai, let's go on the bandwagon. But I think every one of them is now really, excited in what they did. So a lot of fundamentals that we brought in for toxicology of nanomaterials globally, right? It's not in the country, but globally we brought this forward as to what can be done, how can we shift from sort of using electron microscopy to flow cytometry to measure whether the nanoparticles are being taken up or not. And that was precisely thanks to CDRI that we had to do this. I'm just kidding, but I think it was good that we were able to develop these methods for the community, I would say, as such. So I'll just I think it's more. So these are all methods that have been developed to figure out where are the negatives and positive controls, all published. These are all published in the best of journals in the world, and people are now following these as methodologies, and that is very evident, and I'll tell you. So this was an example of zinc oxide which we presented to the world, and this is how it happens, that the particles go inside, they create oxidative stress, lipid peroxidation, DNA damage, blah blah blah, whatever happens. Now the best part was this was published in Toxicology Letters in January of 2009. We immediately got an email from the European Commission that we are looking at this kind of data, and can we have an interview? I said no way, because again I'm a government employee. I had to write to the director, and the director said, well, if you want, you should ask them to send you questions and you show me the answers, what is going out from here, and only then you can answer. That's how it is. So we asked them, and they sent me a list of questions. We sent them the answers, and then they wrote in April itself 1 full page on what we had done in the Science for Environmental Policy. This is within 3 months of you publishing your work, and not in Science or Nature, just Toxicology Letters, which doesn't have a fantastic sort of impact factor. Currently, we have more than 825 citations for this paper, only because of the science that is done within CSIR and for the risk that was supported by the then director or CSIR as a whole, right? As part of the European policy now. Okay. Most of the electron micrographs that you saw, Elizabeth from Canada, she was publishing a book and she wanted them, so she's taken permission, and all the stuff on titanium and zinc oxide is published in the book with permission of the respective publishers. Then came, of course, the bacteria. We worked a lot between electron microscopy and flow cytometry. We developed new methods for that, each of which are now really good. For the students, I'd like to mention, especially when we looked on the microscope, this was really interesting, that the cell was dividing and it still had particles inside, the second generation. And as we moved, we could actually look at flow cytometry and work up to 3 generations of bacteria and still find nanoparticles after the exposure of the first generation. And we kept on going back and forth with CCMB to validate our results with flow cytometry and electron microscopy. So that was fantastic. Yeah, so these are just pictures taken there. And so we were doing Ames tests with salmonella, so we went ahead and did with salmonella as well, whether they are getting inside or not. Of course, a lot of electron microscopy took place, and this became the first paper for detection of or uptake of bacteria in uptake of nanoparticles in bacteria using flow cytometry, and now it's being heavily used. And this was also a paper that became, cytometry doesn't publish cover page articles, but they say it's a featured article, so that's how it was. Right. So the final thing I think was to use in silico, as I mentioned to you, and this we looked at how graphene and graphene oxide, they would interact with a protein. A lot of learning here, and this just for students again. The person who joined me was a gold medalist from MS University Baroda, JNU program biotechnology project. उसमें MSc biochemistry, biotechnology he had done through the JNU program. I said, sir, मुझको मोल बायो करनी है. So I said, till your stuff comes, you learn this, You go to Pune, learn this and come back, which he did in about 3 months' time. Never went back to molecular biology. He's well settled in the US doing what he did. But the best part was that we looked at a biological problem, we looked at a solution that was computer based, and we published in a journal which is a purely chemistry journal. You can't get more interdisciplinary than that. I think the prime objective of this was to look at drug sort of delivery. They were looking at graphene and other carbon-based material that time for drug delivery specific areas, and that's how we looked at it. Over a period of time, we developed the entire range of things at IITR, right from synthesis all the way for all the toxicities, including in silico prediction. We did have a network program of IITR, of CSIR, of which IITR was the nodal. And yeah, the exciting part was that when we did our first international conference 2008, now that is where the timelines come. We started 2006, the first international conference in Lucknow was 2008. I had no clue what was happening, but I thought the only way to learn is to invite the best people in the world. And we did have the lady called Sally Tinkle, who had written the National Nanoscience Initiative of the US, to come and inaugurate the conference as well. And what we came out with was, of course, the people had deliberations and they wrote down what India should be doing moving forward. I think 10, 11 countries participated here. Again, as a scientist, I sent this report for our internal newsletter, and the then director thought it's too much. So he cut it down to 2 pages, but fortunately CSR published it as it is, and it's available on the net. And this was the foundation of the policy for nanomaterial toxicology in this country, or nanopharmaceutical in this country. Of course, this is, so then we went ahead and published our first, in 2011, our first policy document that was released on the guidance of safe handling of nanomaterials. Is all based on our understanding of nanomaterials in the laboratory, how we should have, and that time barely other countries had some of these kind of guidelines in place. So initially we thought we should publish directly, kind of, so they said it should be a guideline, not a rule or regulation. Then as we progress forward, we'll see what to do with it. At the same time, ICMR set up a committee to formulate the nanopharmaceutical guidelines for the country, nanopharmaceutical guidelines, of which I was part as well. And while we were going forward, I think this, we also trained 8 European countries. They came to our institute, we trained them in nanomaterial toxicology, and they went back and established it in their own countries. So that was also remarkable. Yeah, and this is the timeline. So as I said, this is when I wrote the email to CMG. We started off in April. We got the first project, I think, in 2006 or 7. We established this. I think we have to revive it, Prabhat, perhaps, as quickly as possible. The first international conference was held in 2008. We got several projects from European Union and the UK on nanomaterials, and then ultimately CSIR decided to fund NanoShe as a network program 2012-2017. We then published in 2011 our first guidelines. That was this one, and then of course the advanced facility, imaging facility, was inaugurated by the then Honorable Minister Dr. Harshvardhan, that included our SEM and TEM both, along with the confocal facility. I think 2017, because a lot of pressure that everything should be documented and brought forward, especially in the Directors' Conference, what is the impact of what the institution is doing. And so we looked at Web of Science in 2017, and we looked at CSIR's positioning in nanomaterial toxicology. We were number 4 in the world, number 5 was Harvard. 2017, I was awarded the honorary DSc for nanomaterial toxicology, the entire work done in India, in CSIR. And this university that awarded me the honorary DSc, it was the Golden Jubilee, it was the 50th year of their university. The only person till date who's been awarded outside of the UK, right? So I think, again, it's only the work that CSIR has supported. that allowed me to receive this prestigious honorary DSc from them. We of course went ahead and published whatever we're doing in forms of books. We got the National Nanotoxicology Facility at IITR in 2018. The first guidelines was released in 2019, of which I was the author as well as the editor. The second one was on nano-based agri-input and food products. This was a combined effort of CDESCO, ICMR and DBT. And this was sort of taking the things forward from ICMR when we had started way back in 2010-11. And I think we must also understand as an institution that policies or frameworks or regulation comes when we have enough infrastructure or capabilities in place. So when we started in ICMR, there was a concern that if you bring regulation right now, we don't even have the capabilities to test nanomaterials, to characterize nanomaterials, let alone, allow them through the regulatory pathway. So I think that took time, and that is where all the ministries tried to come up with capacity building across the country, and ultimately these guidelines were released in 2019 again by the then Honorable Minister Dr. Harshvardhan. And you have the CDSCO, Dr. Renu Sarup was heading DBT and Balram was heading ICMR, so that's how it got released. And these are the two guidelines available on the DBT website. They're easily downloadable. You can read them, very simple. I think, there's not, I think some 12, 14, 15 pages, whatever they are. And subsequently, we also looked at the European Commission's Scientific Committee on Consumer Safety, and we found some of our papers are now part of this guidance document on safety. The Nano Mission came up with their own guidelines on which they have sort of looked at our guidelines and then taken them forward. Of course, the books have been, these are the people who have actually contributed immensely. They all settled now around the world, and I stand here sort of talking to you on the laurels or the achievements of these kids. But again, I would say when you talk of, it's an investment that somebody made in me, right? So I think it's very important that when you look at the investment being made in you, what is the ROI, what is the return that you give? And this cartoon actually I took from once upon a time from IIT Delhi alumni website, and I thought it was very intriguing that you. So this is what they have written. Something somewhere went terribly wrong, but the way I look at it, because Dr. G.N. Singh was putting a lot of emphasis on AI in healthcare. So I thought that if you started here, and this was the Stone Age, this was the Iron Age, this is the Modern Age. We went back to where we started. Right? Stopped using our brains and stopped, looking at what it is. You rely more on Google than what you think of. So I think human intelligence should prevail. Artificial intelligence should be a tool that you can use perhaps faster as well. But at the same time, I think I'm very in line with what Professor G.N. Singh said, that innovation is the key. So EMI, everything is 100%, a lot of motivation, lot of observation. And motivation comes from leadership, but from within you as well. So I think it's equally important that you feel self-motivated when you're doing things in the laboratory. How you're going to move forward. Good. But at the same time, you have to ensure that you do not lose on your self, because this is the environment, all animal species, and this is the human phase. So if you are not careful, perhaps, again, Dr. G.N. Singh mentioned in his talk in the morning that suddenly the entire campus has become very green. So it's good that you're preserving your nature around you, ecosystem around you. It's very important for us. This is something that I think is equally important. So the leader sort of takes you forward. The science behind it is that there is a little bit of vacuum here, so these birds can fly with lesser sort of input or whatever. And then this thing goes from a V shape to L shape, and they keep flying for thousands of kilometers, right? But the art is when this leader has to move back and allow the next one to come forward. So Rath, thank you very much for moving. Of course, he's left a wet footprint, which is very, very true. It is across the country, he's left a wet footprint in terms of GLP, in terms of genetic toxicology, in terms of the way he interacts with people, lot of people will be motivated in what he has done. I always say there should be an eye for observation, more so in CDRI, because you're dealing with drugs and safety of these drugs. Same photograph, just a bit of introspection, you see the person who clicked the photograph inside as well. Same experiment, everything is same. How you interpret, how in depth the observation is more important, and that is what would lead to good scientific evidence that can be utilized in policy when you come to it. All these policies that I showed you, they have taken the scientific evidence on board. It's not just because me or GN Singh was sitting there and we signed off a policy. It doesn't happen that way. You know, there are stakeholders after stakeholder dialogues and discussion, and only then they come to a point where they are acceptable. Several of these policies, including food policies, come out from IITR as well. So I think it's very important. Again, these are some pictures that Calcutta, somebody is talking this morning, right? So this picture, 6:30 in the morning. For once, I got up at that time. But yeah, so this bird, and those days we had these small cameras with 2 inches of LCD panel. We couldn't see it, so I took it back to my laptop. Still could identify the bird. Then you figured out this is a crow, this is the feather, this is the ray of light coming through, right? So you need to figure out. My bottom line is that observation is the key to success. If you are a keen observer, if you have that in mind, don't worry what is happening, just record what you are getting. I think that then you make sense of what happens afterwards. Don't go, and I think what Dr. GN Singh mentioned, unbelievable the way it happened. GN Singh साहब 1 minute आपको दूंगा बस. 2 minutes? 2 minutes. I'll finish. So, GN Singh साहब came to me, डॉक्टर साहब बहुत जरूरी काम है, 4 लैबों से मेरा MOU करा दीजिए. तो मैंने कहा सर आप इतना छोटा क्यों सोचते हैं, बड़ा सोचिए ना, पूरा CSIR से कराते हैं. क्या बात कर रहे हैं डॉक्टर साहब, CSIR आप बस ये 4 करा दी, मुझे मालूम है, हो नहीं पाएगा. So I said आप 1 minute रुकिए. I rang up Shekhar. I said, you know, I have the advisor to the Chief Minister sitting with me and he wants to have a pan CSIR MOU. Are you willing to have it? He said, why not? So I said, how about we have a meeting tomorrow in the morning so that the 2 of you can discuss and we'll take things forward. So we hung up the phone. He said, I'll come tomorrow at 9:30 in your office. We'll have a Zoom call or Skype, whatever it was, I don't remember. And in the meantime, he spoke to someone in the head office and I got an MOU from the head office in the night. Next day, Dr. Singh and Shekhar had a meeting in my office and they agreed to it. He said, I'm fine, you can take it forward. MOU has already been shared with Alok. I shared that MOU with him. It took about maybe a month or 2 for him to get it cleared through the system and here we are. So I think it is unbelievable the way GN Singh Saab thinks. Again, it was a risk worth taking, but the only state in the country that has a pan CSIR MOU, and you can actually draw a lot of strength from this MOU. We don't realize because it is already existing, you can actually go ahead and do what you want with the government. They've already identified Dr. Singh as one of the nodal people, then the bureaucrats who have been identified. So I think it'll be really good. And the main point, if you see, was related to the pharmaceutical udyog niti, the pharmaceutical policy 2018, which Dr. GN Singh was talking about. So I think it's a win-win situation. So again, everything that you move forward with is Dr. Rath, I could get some photographs of you. I think he has always been a pillar of support when we were organizing workshops at our institute, and yeah, it's been wonderful spending time with him over the years. Very approachable, always standing by you. There was kind of funny photograph here. Alok यार क्या बात कर रही है बताओ. यही रहा होगा यार. I mean, from what I know you, this must have been the only question in your mind. But yeah, that was good. I don't know what I said back to him, but anyway. So I think again, thank you, Dr. Rath, for giving us this opportunity to assemble together and you know, laugh it off and spend a good day worth our time and memories that will sit in our minds forever. And thank you, Amir, and thank you, Radha, for giving this opportunity to again come back to my alma mater or place where I belong and share my 2 cents. Thank you very much indeed. Thank you.
Thank you, sir. We now move to the third talk of the symposium. It is my privilege to invite Dr. A.B. Pant, Senior Principal Scientist at CSIR Institute of Toxicology Research, Lucknow. Dr. Pant is a globally recognized expert in the field of toxicology, stem cell, and developmental neurotoxicology. He's a PhD in biotechnology and a fellow of multiple prestigious scientific academies, including Academy of Toxicological Sciences, USA. Dr. Pant has led pioneering research into human stem cell-based models for assessing neurotoxicity, and not many of us of you would know that his team was the first one in India to publish mechanistic studies on developmental neurotoxicology using human cord blood-derived neurons. More than 150 research publications and extensive contributions. He is a true luminary, and please join me in warmly welcoming him for this talk today. The title of the talk is Development of Human iPSC-Derived 2D and 3D Models for Toxicity Safety Assessment, CSIR-IITR's Perspective. Sir.
Thank you, Dr. Smriti. So, very good afternoon to all. So, after the policy matters which have been discussed by two legends, I am just taking you back to the laboratory where we are just conducting some experiments, but at the same time, Dr. Amir was saying me that you have limited time to speak, so I will just go fast because all of you are hungry. So basically, where is that? Oh. So today we are here just to celebrate the illustrious journey of the Dr. Rath. So no need to say that that he is just leaving a legacy of the excellent research and regulatory toxicology, cancer research, and at the same time the drug discovery. So Dr. Rath, just thank you for giving this opportunity, that because of you we are here and I am just giving this talk. So today just I am giving you just few of the glimpses that actually that in what way that CSIR IITR contributed towards the development of in vitro 2D and 3D models using the stem cells and iPSCs. So since the time is less, so that I'll just many of the slides I'll run through, and this project was started in 2002 as a 10th 5-year plan, and then it is still continuing. So of course this slide is not needed right now because that everybody knows that what is stem cells and how they are working, but I thought I should keep it just to start with the very beginning. So that means perhaps the zygote is the first totipotent stem cell in the body, because from that the entire body is formed. Then the cleavage is happening and that the 3 germ layers are formed, and these germ layers can form every body organs except the placenta. That's why the pluripotency, and that amniotic fluid, cord blood, and the placenta. These are the research sources for the pluripotent stem cells. As we grow, then in the mature adult human body, we can get the stem cells from every organs, and they are either multipotent or oligopotent. So that means these are the pluripotency. And that one question comes in our mind, that why and how these stem cells are different from the rest of the somatic body cells. One thing is that they are uncommitted, undifferentiated cells having the unlimited proliferation capacity, but at the same time they have the capability to differentiate more than 200 lineases if the specific stimulus given to them, what the other cells cannot. So that means the liver cell will produce liver cell, kidney cell will produce kidney cells, while a stem cell can produce any of the cell. So it's the specific requirements if we are giving them. So using that the people exploit these quality and that make the degenerative medicine in that way. So if you look at till 2006, this was the statement that differentiation is irreversible commitment. Once a cell is differentiated into different terminally differentiated cell, it cannot go back. But in 2006, there was one scientist, Yamanaka, who first time proved no, terminally differentiated cell can also be reprogrammed and make a new kind of stem cell that is a induced pluripotent stem cell. So that means from that induced pluripotent cell, the different kind of organs or tissues can be made. So this was the breakthrough journey of Yamanaka, which people are using all around today. So that means, what is the advantage of this pluripotent stem cell above the stem cell? Because that means if we are getting a somatic cell from the patient and making it a iPSC, so that means a patient derived iPSC, stem cell and different kind of cells can be made for the drug development model or disease model system. So the plenty of number of cells and the ethical dubious is minimum, because we are taking a simple leftover tissue. Maybe after surgery we are taking some tissue which is not of use, that can be utilized for the making iPSCs and for the different organ system and all this. So what IIT are doing in the stem cell and iPSC area, as the name of organization spells out toxicology research, so that means our domain is in toxicology. So the primarily when we started working with just to make a developmental model or an in vitro model of the developmental neurotoxicity and neurotoxicity. So I think there is no need to tell that what is developmental neurotoxicity to this audience, but just to let that today that it is well recommended or well documented in the literature that the drugs and chemicals which can induce the toxicity in developing fetus brain cells or children at early stage of life till the blood brain barrier is not developed are more vulnerable to concentration which is non-toxic to the adult's brain. I will just simply give an example of that. We all are using the mosquito repellent. So in a mosquito repellent that pesticides are there. So that concentration of pesticide is not good enough to induce any adverse effect to the adult's brain. But if a lady who is in the family way is getting the exposure of the same pesticide, so what the concentration is going through that lady to the fetus, that is good enough to induce the degenerative damages into the developing fetus brain. So that way, and at the time of birth, that child will absolutely normal, and that abnormality will appear at the age of 6, 7 or 8 years time. So we will not aware that why and how it has happened. It has happened because of the exposure which she or he got at the age of gestation period or LT stage of life. So that prior to put any product into the market or a drug, because in every year we are putting approximate 8 to 9,000 chemical in the kitty of chemicals to the world, and that safety analysis of long-term safety analysis of every chemical is not possible at all. So that how we know that this product should be safe to give a pregnant lady or not, whether the changes what will come that is reversible or not reversible, as such no model system was not available. So with this thought we started working in 2002 with this stem cell, and that we just started taking the stem cell from the cord blood, because that is a liquid tissue. The dragging out the stem cell was comparatively easier, and that ethical dubious was less. Even it took 2 years time to get the ethical clearance for this cord blood, and in the last meeting I fought with Dr. S.S. Agarwal, okay sir, when you will throw it, I will catch it and I will use it. Nothing will happen to fetus or baby, because we will just take blood after clamping. So then he just agreed to this, and then we started working. So what strategy we had adopted in IITR, that there are two strategies. One is to develop new experimental models, alternative models. We will do the science, and then we will try to incorporate those models as a acceptable model for the regulatory purposes. Second strategy was that models which are already available and adopted by the OECD and other regulatory agencies, those should come to CSIR or India, and then we have to utilize them for the purpose. And ultimately all these models will be incorporated in the GLP, the battery test system. So if you look at, we generated lot of data and try to convince the DCGI and other agencies, please incorporate these models, these validated models in the drug development system. And in 2023, then in the new drug and clinical trial regulation, they have made 175th amendment, and there they have incorporated all these cell-based assays as a first year of screening for the drug development. And now this is a part of document, and they are accepted models, and the people are using as a first year of screening for the drug development, because everything cannot be screened into the animals or to the other systems. Okay. So with this, we have started with the developmental neurotoxicity. So just show you the glimpses. That first task was to just purify the stem cells. So first, where is this? So sorry. Marker किधर से आएगा? Okay. Okay. So first time when we just purified then those cells, so we got around 90% purity of the stem cells by using the RoboSap, that is the electromagnetic separation unit, and it was the first in India and in IITR in 2007. So we bought it and we used it, and thereafter using the different permutation combination of the growth factors and everything, we reached to the purity of around 99%. And then the other task was how to just make that they are purified, they are physiologically functional. So n number of parameters we have analyzed, and finally we just confirmed it. Yes, what the stem cells we are dealing, those are purified population of the one type of cells, and those are hematopoietic stem cells. So that time the purification definition was CD133 positive, CD34 positive and CD45 negative, and then with the all purity that we could get this. Then we went through this, yes, then we allowed them to differentiate into the different neuronal subtypes and glial subtypes. That time no protocols were available. It took 2 years time to make the different combination permutation of the growth factors, and finally we could reach to this, and that this is a morphological differentiation. Then the different kind of more than 100 markers we have analyzed, and this is just I have put it one, that is the dopamine DA-D2 receptors, because when we just grow them dopaminergic neurons, so that means all dopamine receptors will just concentrate in the nuclear part, so that these kind of typical 2 rings will appear in the antibodies, and that shows that yes, the purity of the cells were there, and we are making the dopaminergic neurons. So we have made the dopaminergic, GABAergic, cholinergic and all sort of differentiation we have made along with the glial differentiation to those cells. Thereafter the major challenge with us was, because if you look at our body is not neutral, we are electrically charged, and the electric current in the brain cell is a greater in magnitude than the other rest of the body cells. So what the cells we were growing in this culture medium, whether they are physiologically functional or not, or electric current is there or not. So that was a big challenge. So for that we went through the all electrophysiological experiments. उसका इसके बीच वाले में क्लिक करना है जरा, मे बी अगर if it is working, कर रहा है काम नहीं. So basically this is the electrophysiology experiment where we found that yes, so basically with this experiment we just confirmed that yes, what the cells we are growing in the culture media, these are having the actual accurate electric current what we have in the brain cell in the body. So that was, we confirmed that yes, the cells which are growing in the culture medium are actual physiologically functional neuronal cells, and those are working this. So starting from the stem cells, from the umbilical cord to the neurologically physiologically functional neuronal cells, we have grown them. So lot of things we have done. So next slide please. So finally that we could just make out this a 2D model system for the developmental neurotoxicity, because we exposed these growing cells at various stages of maturity with the known developmental neurotoxicants, and the look at which specific genes are getting up and down regulated at transcription level, then at protein level, and then post protein level, that is in the functional level. And then we just added the neuroprotective agent as well to confirm whether the changes are reversible or not reversible, and whether these changes are chemical specific or the monotonous changes. So with a variety of chemicals, drugs we have used,
And after that, because the entire 40 weeks of gestation period is mapped now in terms of physiological activities. If we just take the amniotic fluid of 14th week, we know that what particular kind of hormones and growth factor should be there in the, if suppose that some hormone is missing, so we could just identify that the gene responsible for producing of this particular hormone is located in the chromosome number 14, locus number this. So that means we can identify and we can just rectify it. So in the same fashion, when we know everything, so doing these experiments starting from all through the developing phase, what the set of genes we have identified, that we have put it in a chip, and then this chip is now being used to evaluate the new molecules which is coming towards the market for preclinical studies and then so on. So this model has already been approved and adopted by the OECD as well in 2017. OECD has remarked this, and it is in the final stage of the draft stage. So probably in a couple of years time, it will be a guideline from the India itself. Then we realize that our body is not 2D, because what the chemical we are ingesting or we are being exposed, that is being taken care of by either maybe multiple organs working together or that some tissues together. So that means we thought of that just making a model of 2D system with the purified population in flask will not serve. Then we just went through this development of 3D model, organoid model, spheroid models and other models for drug screening. So because the molecule showing, if you design the 1000 molecules synthesized in milligrams, so we can funnel down them using these 2D model system, and molecules showing high efficacy and safety in the 2D model system can go directly with the 3D models instead of the animal system. So this intent, we have started working with this around 10 years back, and these are just some glimpses that how we grow the iPSCs, human derived induced pluripotent cells. And after that, started with the marker analysis, and then amyloid body formation. That means like a amyloid, 3 germ layers are formed. So that means hundreds of markers we have. I finally just, because time is not permitting me, so just I am showing that actually what happened in the last. So finally we can just grow the ball of meats in our culture medium. They are physiologically functional and they are expressing all marker. Electric current was there, and now the question was, okay, electric current is there, markers are there, everything is there, whether those will work in the body or not. Then we just went through this experiment with the nude mice, and in nude mice, when we injected the cells, and it took initially 3 months time to get these kind of teratoma formation and other things, because we was really with the tears in eyes. For one fine morning my student came and, sir, nodules are there. Then we jumped on and immediately went to the animal house, and they see that yes, they are there. So it shows that yes, what the cells we are growing, those are actually physiological functional and they are working inside the body, and now we can utilize them for the drug screening or for chemical screening. So with this, I think this video will not work, I will just skip it. So what the other model we are doing, that using this protocol, because now protocol was established, we can just make the 2D and 3D model starting from the iPSC and the stem cells. Then we just took one disease for which I got the Indo-Brazilian project as well. So amyotrophic lateral sclerosis. So it is a disease of motor neuron. So after diagnosis, the patient will survive only 4 to 5 years. As such, no treatment is there. So first thing is to make a model of that which is mimicking to the human system. So that means a high throughput screening system can be developed for the development of the drug. So that means for this also, just I will not take much time, because starting from the patient fibroblast, we just reprogram it and make the iPSC, patient derived iPSC and the normal iPSC. So based on that iPSC, then we differentiated them into the motor neurons and astrocytes. It took around 48 days protocol. So finally we could able to make these motor neurons and astrocytes from the normal iPSC as well as the patient derived iPSCs. So both the models were available. Then we just validated them that yes, from the Cas9 technology, using this technology, we made the transfection for the TDP43 and SOD1, two specific genes which are prominently involved in the ALS disorder, so that we have made one purified population of the organoids from the iPSCs, normal iPSCs, and one from the patient. So now these models, that these 2D models, as well as then we went for this 3D model development. So that means we made a spheroid model, organoid model for the ALS disease, and now this model is extensively being used for the screening of the drug molecules which are directed against the ALS and other neurodegenerative disorders. So now we are just because ALS start from the motor neurons in the brain and connect to the spinal cord, and that spinal cord is other end attached to the muscles because it's a muscular disorder. So we thought of, instead of just making organoid, we should go one step ahead to that, and now we are making assembloids, and this assembloid is also accepted by the DCGI as a model for the pre-screening. So at IITR, sorry, sir, we are making the assembloids of the, basically we are making organoids of the cortical organoids, the cortical region of the brain, motor neurons, and then other spinal cords, and then fuse them. And this is a few photographs of these assembloids what we have made in our laboratory at IITR, that those two kind of organoids, then they work together as a assembloids, and that transportation or migration of cells from one side to other side has already been established by the transformation, because we have made a transfection with the GFP tagging. So that means from that migration of the cells can also be easily tracked. So that means this model is almost done, almost. So that means very soon that we are just going to utilize this model for the purpose. So this is this project where that this stem cell we have used. There was a multi-CSIR institutional project started during the period of Dr. Mashelkar, where that the idea was just to screen the natural and semi-synthetic molecules and to get the patent of that. Dr. Nath was coordinating in this project from the CDRI. So the job of IITR was the in vitro screening for this, basically dopamine receptor, cholinergic, benzodiazepine and serotonin. So that IITR was supposed to do the in vitro screening in the synaptic membrane of the brain. For this, we have sacrificed thousands of rats and mice. So in the mid of the project, we thought of, instead of sacrificing animals, if we could just make it from the human iPSC derived neuronal cells and just use that synaptic membrane, whether it is working or not. So just we converted those stem cells and iPSCs into the different brain region neuronal cells, dopaminergic neurons, cholinergic, GABAergic and benzodiazepine, and just look at the comparative analysis, and what we found, yes, that are actually giving the mimicking results what we were just getting from the synaptic membrane of the rat. So that means from the mid part of that project, rest of the project we have conducted using those in vitro cells only without any rat model. And that system is now being used by everyone. So that is another important approach that we had used. In 2000, no, but sir, no. Yes, the CDRI was supposed to do the in vivo studies. IITR was supposed to do in vitro analysis. Sir, but for in vitro receptor binding, we are getting synaptic membrane from the rat brain. That was the issue. Sir, 27,000 molecules IITR has screened. 27,000 molecules. So no. Hi. No, test was only 6 receptors. So. Yes, yes. Yes. Yes, madam, just 2 minutes I'll finish. Sure. So basically in 2014, that the animal studies for the safety studies of cosmetics was completely banned in India. So that this was a pressure to establish the model system, in vitro model system, on which that we can screen the preclinical screening of the cosmetic product. So that IITR took a challenge, and that the EP skin and EP ocular model that we have made. So this is one picture that where we have, this is our indigenous 3D EP skin model what we have developed at IITR, and otherwise also EP ocular we have developed. This is histopathological examination, but the membrane what we have made at an IITR, and this is epidermal skin system. So that means all this system at IITR we have made, and the beautiful part is that what actually we were getting from the outside, those were the membrane or the skin of the European population, of the US population, where the skin type is different from the entirely from Earth. So indigenous, our diversity, our genetics and everything, so response will be more similar. So probably last slide, it is scientific last slide. So that means the other challenge what we took in last 2 years time, because a model for cosmetic toxicity, that if you look at, these are the two regions where the stem cells in the hair are there, and this is the original picture of the growing hair in the culture medium. So that means that we have done this, we have made this scaffold model for the hair testing for the cosmetic products, and this is the results. So at day 0, this was the growth. So that means if you look at that, with a positive control there is no growth, and at day 6 there is a growth. So that's something that we have just developed there in IITR, and now for the cosmetic we have an entire range for the 2D models and 3D models for hair, everything is there with us. So this we have recently initiated, because that multiple organ system on a chip. So that means this we are just in a process of establishing here, because that for this what the people are doing across the globe, we took a little bit different way with the precision that we will just make the continuous flow of the medium with the growth factor. So that means the accumulation of the toxin should not be there. But ideally, because in a static system that the accumulation of toxicant will be there, so that means more aggressive results made there. So that means in our system that we have a circulated system, so that means bioaccumulation is not there. So that means for this region, if you look at that, one is that continuously injecting, that organoid is put it there, one is the inlet and one is the outlet. So that means this is indigenous system what we are trying to make it so precisely. So that means every organoid put it on the chip will get a continuous flow of the medium where the biomagnification of toxicant will not be there. So we are in the process of standardizing it. So probably in the next 6 months time we will be having the data sufficient to that. Okay. So basically this is just I have put it here for this. Deliberately I put this. This is one book, the Dictionary of Toxicology. This is a 1100 pages book. So basically in the dictionary you will find the one meaning of the word, but here you will get minimum one paragraph of everything. So 3500 words are there. This is 1100 pages book. So that means this is the gathered information at one place. So that means if somebody wants to look at in the toxicology, so that information is there. So that is all this. Thank you. Thank you so much.
Thank you, sir. Now it's time to felicitate our honorable guests. I will request our director, ma'am, Dr. Radha Rangarajan, to please felicitate our chief guest, Dr. G.N. Singh, our guest of honor, Professor Dr. Alok Dhawan, and Dr. A.B. Pant, our distinguished speaker for today. I now request our guest of honor, Professor Alok Dhawan, to Dr. Aditya B. Pant, may I request you to please come to the dais. With this, we come to the end of today's program. Once again, on behalf of organizing committee, I thank you all for joining us on this meaningful occasion. Thank you all. Please join us for lunch and have a nice day. Thank you.